So, good morning to all our colleagues from US and good evening to participants from India. Welcome you for this Immunogenetics virtual webinar. And I now I give the mic over to Dr. Keval Krishan, who is the president of the Indian Society of Heart and Transplant, to give a welcome note and say a few words.
So don't want to take more time. I'm grateful to everyone who accepted to deliver a lecture and tell a lot about immunogenetics, which is anyway the surgeons are, they are poor at. Basically, we should accept that we are very poor at in this area of medical science, that immunogenetics. So I think it will increase our knowledge and improve our outcomes also. And at least we will be in very much connect to our connection with our physicians, those who are taking care of our lung and heart transplant patients. And the newer 2-3 things which I would like to mention, that we are coming up with a new website of INSHLT with a lot of new contents, which is state of the art, and few surgical videos, educational material, and a lot of other things. Some companies, those who are doing certain tests, which most of the surgeons, they don't know, we will put them on the board so that they can go through that and see where the nearby labs also. Another thing we have on WhatsApp, the most of is the scientific discussion, which is really good and which is confined. Our WhatsApp group of the society is confined to the knowledge as well as to the scientific program. So we are also coming up with this Twitter, INSHLT Twitter. So, and the another thing is Dr. Sundar, he from Chennai, he's organizing one heart failure society, Association of India's meeting with in association with INSHLT in mid of the February. And IACTS, the Indian Society for Cardiothoracic Surgeons, they have given us one hall for 2 days completely dedicated to heart and lung transplant and MCS. So that will also give us a lot of, we'll connect all the people, those who are interested in this field, and give a lot, put a lot of light on in this subject, so that understanding of our doctors will be much better, and we can give really good results and promote the new people, so that we give inspiration to the people that this therapy is really good and working in this country. Thank you very much. I will not take much time. I think we should go ahead with our speakers to deliver lectures. Thank you very much.
Thank you, Dr. So I just give a brief about the expert panel. So we have a lot of our distinguished panel here. So first is Dr. Jignesh Patel, who is very well-known transplant cardiologist from Sadas, Chennai. So we welcome you, sir. Then Dr. Professor Kumud Dittal, who was a long stint in Australia in Sydney and Melbourne. So welcome, Dr. Kumud.
Thank you.
Dr. Gokhale is a senior cardiac surgeon from Hyderabad, and he's the ex-president of the Indian Society of Heart Lung Transplant. Welcome. And then our other panelists, Dr. Palak Shah, I can see him from Fairfax, Virginia. Welcome, Dr. Palak.
Thank you.
Dr. Savesh Pal Singh is a senior cardiac anesthetist from All India Institute, which is a premier cardiac center in New Delhi, India. Welcome, Dr. Savesh. Dr. Srinivasan, we asked him to be here because he heads one of the very big HLA typing labs in Chennai. It's one of the few in India, and they have a big database for especially bone marrow HLA typing. So we asked him to be here. Dr. Uma Kanga is head of the Transplant Immunology and in All India Institute, New Delhi. She'll be speaking later. Dr. Dolly from CMC, Christian Medical College, Vellore. And Professor Jasmeet, she is from Lalpath, one of the biggest labs. So I think Dr. Howard Eisen possibly would be joining, possibly in a short while. So the first speaker would be Dr. Ju H. Kim. He is assistant professor in advanced heart failure and transplant cardiology in Houston Methodist Hospital. And he's got a significant experience in the non-invasive immune monitoring for heart transplant tests like AlloMap. He was also part of the Cargo 2 trial. So Dr. Kim, I think without much ado, probably you can start your presentation and we can take the questions as after the end of the session.
Well, thank you very much. And thank you very much again to the organizers for the invitation. It's really an honor to actually be able to contribute to a webinar such as this with really a distinguished panel of speakers, certainly experts around the world and certainly in the country that are far more experienced. And I see it's good to see Pollock's face there. Again, a colleague of mine who's taught me along quite a bit along the way. So it's really an honor. My name is Ju Kim. I'm a cardiologist here at Houston Methodist in Texas. And as I'm the first speaker, my aim is to just introduce the concept and go over a clinical application. And I'm going to just do this by sharing a case. So my objectives are just to review the use of gene expression profiling and donor-derived cell-free DNA for acute rejection monitoring after heart transplantation. And I'm going to give you a real-world application of combining both of these tests and hopefully spar off a discussion trying to figure out what is the clinical role at the present time and in the future for potentially reducing myocardial biopsies and tailoring management decisions. Just as a disclosure, I have served on advisory boards and a consultant for CareDx. As this panel is very familiar, the endomyocardial biopsy still remains the gold standard for rejection monitoring. And the ability to sequence the human genome really is what led to revolutionary progress that allowed us to really assess the relationship between the genomic and transcriptomic and proteomic variations and see what the relationships are to clinical phenotypes. And going back to 2001, and the Cargo investigators really aimed to identify and validate this gene expression profiling test to identify those heart transplant patients who had a low probability of moderate to severe acute cellular rejection. And this test was developed against the reference phenotype based on the endomyocardial biopsy. And Cargo and Cargo 2 were studies that really developed and validated the test characteristics in multiple centers in the US and across Europe. And really it demonstrated that AlloMap, or the gene expression profiling test, was able to differentiate the presence of acute cellular rejection from those without. And again, as I'm sure this panel is more familiar, the AlloMap, or the gene expression profiling, is reported as a score from 0 to 40, and the contribution to the score is dependent on the expression of candidate genes. And they're looking at the mRNA levels of 20 genes, 11 of which are the reporting genes representing these multiple domains and molecular pathways, and 9 of which are control genes that are used primarily for reproducibility and standardization purposes. And the score that's reported is a composite of all 11 of these genes. After Cargo and Cargo 2 really developed the test characteristics, it was really the IMAGE trial that was done in 600 patients, 13 centers, and looking at heart transplant patients beginning at 6 months to 5 years after transplant. And it was designed to test whether a non-invasive rejection surveillance strategy using this technology is not inferior to the traditional endomyocardial biopsy-based strategy. Composite outcome here was rejection with hemodynamic compromise, graft dysfunction, death, or retransplantation over a 19-month follow-up period. There are really no differences in the baseline characteristics of the patients, including their immunosuppression regimen. And this Kaplan-Meier curve sort of show that not only is there no effect on survival, there was no difference in the primary outcomes with regard to rejection, hemodynamic compromise, or graft dysfunction. And so the IMAGE trial really did show that clinically it was able to not only distinguish those with acute rejection versus not, but it was not inferior as a strategy for monitoring these patients who are at low risk of moderate to severe rejection. And this study was able to actually prove that we were able to reduce the number of biopsies done, and there was also improvements in patient satisfaction that were reported at the time. After image, the e-image study, which I think Dr. Jay Patel can speak to a lot more detail, as he was one of the lead investigators in this, enrolled 60 patients much earlier than the 6 months, starting at 55 days post-transplant, and looked at the similar sort of low-risk patients who were on low doses of corticosteroids, clinically stable with a preserved allograft function by echocardiogram, and who had really low risk of rejection, and looked and demonstrated similar findings to the image study that showed the surveillance beginning at 2 months post-transplant with this gene expression profiling was not inferior to an endomyocardial biopsy-based approach. One of the limitations of this gene expression profiling, or the AlloMap test, is based on its positive predictive value. As you can see from the data extrapolated from the Cargo study, the score as it goes lower down, the negative predictive value goes up, but as the score goes up towards 40, the negative predictive value still stays really high, but there's poor positive predictive value. Furthermore, the gene expression profiling test can be affected by various clinical factors that were commonly seen in transplant patients, whether it's the dosage of the corticosteroids for maintenance immunosuppression, whether the patients have gotten recent blood transfusions. There's also more data in a paper that just recently came out in HLT looking at the impact of CMV infections on causing false positive gene expression profiling scores. And more importantly, this test was not validated in antibody-mediated rejection. Nonetheless, the last guidelines that we have from the INSHLT in 2010 did assign a Class 2A level of recommendation for using AlloMap to rule out the presence of 2R or greater ACR in appropriately selected low-risk patients between 6 months and 5 years after heart transplantation. And it's interesting to note that, again, the quote-unquote gold standard carries the same level to a recommendation with a level of evidence C for using endomyocardial biopsy to surveillance for acute rejection post-transplant. So with this, AlloMap, or the gene expression profiling test, was widely adopted for acute rejection surveillance by certainly centers around the country, including this center here. And the high negative predictive value is something that was taken advantage of to try and rule out the presence of acute cellular rejection in the low risk population. Which gets me into the case to hopefully spark some discussion. This is a 59-year-old patient with a history of non-ischemic cardiomyopathy who underwent heart transplantation towards the end of November in 2019. Pre-transplant, his PRA class 1s and class 1, class 2 were zeros. And at the time of transplant, his cross-match was negative for T cell and B cell by flow cytometry. Here at Methodist, we do not routinely induce all patients. He only gets steroid only, especially for low-risk patients. And he was maintained on a regimen of tacrolimus, mycophenolate, and prednisone. His rejection burden early on was a 2R rejection at week 4 with no significant hemodynamic compromise that was treated with outpatient steroids.
Hi, yes.
I would note this particular patient's biopsy was also technically challenging. He had a right IJ DVT. His left IJ was pretty stenotic as well. And all of his biopsies were done by a femoral approach, but even then, his right atrium was quite generous. And there were times during the biopsy, even through the femoral approach, that we had to use ICE or intracardiac echo to guide the bioptome. And it was a challenging procedure for all of us in the group. And beginning at week 8, or 2 months after transplant, we initiated non-invasive surveillance, and this is the first AlloMap score for him to try and establish a baseline, and the score came back as a 29. And throughout this time, his echocardiogram showed preserved allograft function with an EF in the 60s. By month 3, he came back for his routine follow-up. He was just reporting some mild fatigue, felt like he wasn't quite getting back on his feet. But otherwise, he was doing well and didn't report any palpitations or lightheadedness or any signs or symptoms of volume overload. And we continued with his non-invasive surveillance, and his AlloMap score came back as a 37, which was a rise from the 29 that we saw just weeks before. So we did bring him into the lab, and here his hemodynamics. His right atrial pressure was 10, his mean PA pressure of 18 and a wedge of 13. His fit cardiac output was preserved at 5.4 liters with an index of 2.6. The echocardiogram also showed normal allograft function with an EF still in the 60s. Surprisingly, however, his endomyocardial biopsy specimen was read as a 2R. Now, in full disclosure, that is not his particular sample. I just picked an image for reference, but it did show multiple foci with cellular infiltrates concerning for a 2R rejection. At this time, his CMV PCR was negative, and we did send off his HLAs, and he did not have any class 1 or class 2 DSAs. And the complement or C4D fixed, stain on the same sample was negative. So at that point, we did treat him again with another course of steroid bolus. And he was actually managed as an outpatient even at this time. Now, month 4, if you remember, his transplant was towards the end of November, and month 4 got us to the end of March. And I'm sure most of us on the call are aware, but the end of March was the time when we were looking at this type of a graph for the coronavirus infections. And it represented a big challenge for us, and just because of the unknown of how we should be managing these patients. Should we even bring them into the clinic and risk exposure? And there was lots of discussions about how to try and minimize not in-person visits, let alone quote unquote elective invasive procedures that may require hospitalization. So we got together as a group and reviewed all of our transplant patients, and we tried to prioritize non-invasive surveillance methods using this technology in appropriately risk stratified patients. And month 4 through 6, we did maintain his non-invasive surveillance using the heart care tests. And I'm just showing you the AlloMap scores here. But we were able to use this to try and avoid biopsies by doing virtual visits, doing remote laboratory monitoring for his immunosuppression drug levels and organ function, and the echocardiogram. Now, this does show that his AlloMap score does go back up above 35 going into month 6 of his post-transplant course. And even through this exact period, we were getting more and more familiar with this particular test, as well as the next component of non-invasive surveillance, which is a donor-derived cell-free DNA. Again, we have other experts that are very familiar with this technology, but we all have cell-free DNA circulating in our bodies from nucleated cells. But in the transplant patients, a fraction of those will be from the donor organ, should be some sort of organ damage, whether it's apoptosis or cell necrosis. And this is taking advantage of the single nucleotide genotyping information that is able to differentiate those polymorphisms from those of the donor versus the recipient. And again, early data showed that the cell-free DNA fractions decline very, very quickly post-transplant, as shown in panel A. The inset shows that within, I'd say, 3 to 5, and by certainly the end of week 1, the donor-derived fraction of cell-free DNA decreases quite dramatically. And then panels B, C, and D show that the derived cell-free DNA fractions do increase at the times of acute rejection, whether it's cellular or antibody-mediated rejection. And interestingly, I think one of the points of discussion and one of the areas that we're going to learn a little bit more about is that the fraction of donor-derived cell-free DNA seems to precede the actual diagnosis of the rejection. And the DOR study, again, led by Dr. Cush, it showed that in the quiescent population, in the reference population for our transplant recipients, the donor-derived cell-free DNA concentrations are very low, and the median level being 0.07%. Not only is it low, but it tends to stay low even months after heart transplantation to at least 2 years. And the DOR study was the one where we kind of showed us that this particular test was able to distinguish between the presence of rejection versus not. And again, not to harp on the details, but it was also able to detect the presence of not only cellular rejection, but antibody-mediated rejection in these patients. So what we're doing here at Methodist is using this HeartCare test, which is a combination of this AlloMap or the gene expression profiling test, as well as the AlloSure or the donor-derived cell-free DNA test, in combination by sending off this HeartCare. And there is, I believe, yet to be published data from, I believe, Cedars that looked at the test characteristics of each of these studies individually and combined, and I believe the test characteristics were much improved when the two tests were used in combination. One of the things that we had trouble with clinically early on is what to do with this spectrum of the test results. Now, even in the DOR study, I'm sure most of you have seen this figure, over 50% of the patients had the results of a low AlloMap and low AlloSure. 6% had both high in AlloMap and AlloSure, but there was this group in the middle there, and 26% whose AlloMap score was high, but in low AlloSure, and the 11% whose AlloMap was low and AlloSure is high. And ultimately, we struggled with this too, the cohorts in the middle with the discordant results. And even looking at some of the guidance that came from the steering committee, of which we have members on this very call, it's easy when the AlloMap and AlloSure are low, it's easy when they're high, but there's lots of different considerations to evaluate when there's discordance. And getting back to our case, one of the reasons that we felt comfortable avoiding biopsy and using this non-invasive surveillance technique even for this patient whose AlloMap score returned back up to 35 and stayed above 35 throughout was the fact that the complementary test below, the AlloSure test, showed that the cell-free DNA fraction was below the lower limit of detection throughout. So even though this patient fell into this 26% where the AlloMap test was reported as high, but the AlloSure was low, we felt very comfortable with the fact that there's no evidence of serious graft damage in combination with reviewing his immunosuppression regimen and seeing him clinically, either virtually or in person, as well as monitoring his echocardiogram to avoid endomyocardial biopsies. So this is one of the topics that I think we are going to learn more about as we go, and we're looking to share our single center experience looking at this clinical implications of what to do with these discordant AlloMap, AlloSure test results. And ultimately, I believe as more experience is gained throughout the field, we'll be able to give some good guidance of when it is that we need to worry and when we should actually go back to doing a biopsy, and more to come with that. And I'll just finish by kind of sharing what we do here at Methodist. As you can see, and we have representatives from multiple different transplant programs, even in the US on the call, but we are a fairly biopsy heavy program, and we biopsy weekly for the first 4 and every 2 for the next 2 months. And then, we do monthly up until month 6. And so with incorporating this non-invasive technology with both the gene expression profiling and donor-derived cell-free DNA, we're incorporating those tests early on. And we're hoping to review our data and review the data from the field to see if we can not only reduce our endomyocardial biopsy procedures and hopefully improve our patient satisfaction, but and the aim is to try and learn more about the field to actually tailor our management decisions. So the conclusion is that the gene expression profiling and donor-derived cell-free DNA have complementary roles in surveillance after heart transplantation. We are not using this in isolation. We are using it in combination with the clinical picture as well as the echocardiogram. Additional data are emerging to understand what factors are that influence these results and their clinical applications. And what we learned is that clear institutional protocols that minimize individual physician level practice variations with mechanisms in place to monitor not only patient-specific, but center-specific data are important to understanding this in the appropriate applications. And with that, I'll end. And thank you again to the organizers for the invitation and the opportunity to share this data with you.
Thank you, Dr. Kim, for a very wonderful presentation. Before I go to the next speaker, I would just like to introduce to the panel four people from who have joined us in the expert panel. One is Dr. Vasanthi Ramesh. So she is head of our All India National Organ Transplant Organization called NATO. She is the equivalent of UNOS in India. Then I would like to welcome Professor Howard Eisen. We are very pleased and honored that he could join us. Professor Eisen, for the audience, doesn't need any introduction for the international kind of experts. He is one of the founder members of the International Society of Heart Lung Transplant. He is a member of the board of INSHLT, and his achievements are legendary in the field of transplant immunology. He is one of the gurus of transplant in the world, actually. So thanks, Professor Eisen, for joining us. Dr. Sandeep Seth, I would like to welcome him. He is Professor of Cardiology and Heart Transplant in All India Institute, New Delhi. And Dr. Suresh Rao is head of Transplant Intensive Care and Anesthesia in MGM Hospital, Chennai. So he's also there as part of that. So next speaker would be Dr. Uma Kanga. She is Professor, Department of Transplant Immunology and Immunogenetics in All India Institute of Medical Sciences, New Delhi, the premier institute in India. She is a renowned Indian expert in HLA studies, tissue typing, and organ-specific HLA registries. Has also been involved in developing the Asian Indian Bone Marrow Registry. She's an expert in flow cytometry and been awarded by multiple organizations all over the world, including Japan and UK. So we welcome Dr. Uma for your presentation.
At the outset, I would like to thank the organizers for inviting me to share my experience. Can I share my screen now?
Yes, ma'am.
Is my screen visible?
Yes, doctor, it's clear.
Thank you so much. So this is my institute from where I work since last 2 decades. This is the Department of Transplant Immunology and Immunogenetics at All India Institute of Medical Sciences. I would just like to first acknowledge the pioneers who were awarded Nobel Prize for organ and bone marrow transplantation. And these are the ones who have been awarded Nobel Prize, but we all know in this society that the heart transplantation contribution by Christian Barnard, and then in India, Dr. Venugopal started the pioneering work in August 1994. And now we have many transplant centers contributing hugely towards this program. So the main purpose of having immunogenetics background and immunology studies along with the transplant is that we want to avoid post-transplant complications. And because of immunosuppressive drugs, there's a lot of toxicity, infections, and malignancy. And all of this, we need to just find some biomarkers that can avoid this hurdle of acute or chronic rejection in solid organ transplantation. So whenever a transplantation happens, there is bound to be an alloimmune response because we have an allogenic organ placed in the body. And how the immune system reacts is like, for those of you who are familiar with cricket, I would like to say that it's like the MHC serves the purpose of a bat. Once the antigen is presented, like the ball is presented to the bat, the antigen is presented by the MHC, and that's how the T cell receptor looks at the T cell, looks at the antigen, and then generates an immune response. What happens in the organ transplant scenario is, oh, the slides are not moving forward.
Just click on the screen once with your mouse.
Okay. Yeah. So in organ transplantation, there is actually a two-way allorecognition when the immune system takes over. So first is the direct allorecognition, wherein the antigen is presented by the host MHC, and then gradually over there is an indirect allorecognition. So I'm giving you an example of a kidney. However, the same thing applies to all the solid organs, that the initial site of encounter is the foreign MHC occurs in the secondary lymphoid organs, and gradually these recipient T cells are presenting the organ. But when we move over a period of time, there is an indirect allorecognition that the host MHCs will capture the donor antigen and present to the immune system. So in the organ transplantation scenario, we need to handle both the direct as well as indirect allorecognition. And we should keep this in mind because I'm going to be talking in future about in few minutes about the indirect allo recognition and the software that we can use to predict this allo recognition. So basically, when there is a lot of allo recognition, these alloreactive T cells will migrate to the grafted organ, and because of their transmigration, they will go and attack the donor organ. And we need to have some mechanisms wherein we can control this alloreactivity using immunosuppressive drugs or some biomarkers that can help us predict that when this alloreactivity happens, how do we lower the alloreactivity. So basically, when an organ transplant is done, so we have to be very careful that the best thing to do is that we have genetically matched organs. And for that genetically matched organ, the main gene system that we look at is the HLA genes that are located on the short arm of chromosome 6. We do match for the class 1 A, B, C, and the DRDQ usually. Now, this system is highly polymorphic and has a lot of immunologically relevant antigen presenting molecules. Therefore, the role in solid organ and bone marrow transplantation. And this system is highly polymorphic, means you can look at the number of alleles that we know of. There are about 28,000 alleles. And we need to have technologies that can be able to detect all of these alleles. And this number is hugely increasing because of the technology advancements in HLA typing. So if we look at the structure of the HLA molecules, majority of the polymorphism lies in the peptide binding region. So that is the alpha 1 and alpha 2 domain of the class 1 molecule, whereas the beta 1 chain of the class 2 molecule is where all the polymorphism is. And this region is what is going to be presenting the alloantigen. So when we try to look at the cell surface, these are lots of HLA molecules that will be expressed on the surface of any human recipient or donor's cells. And there will be a number of HLA molecule because from the 2 chromosomes we get all the HLA A, B, C, D are expressed. So because of their nature of being polygenic and polymorphic, 2 individuals can differ from each other by the number of HLA alleles that they share, because certain genes will differ at the class 2 or at the class 1. And on the basis of that, we identify that individual, how much is the compatibility between a recipient and a donor. And for example, I can show you that if you look at the cell surface, if at the lower panel you see that the individual is homozygous, there would be only few number of HLA alleles on the cell surface. Whereas on the top panel, if you see that the individual is heterozygous, and on both the chromosome 6, there are a number of HLA alleles, then the number of antigens that the cell will express is going to be much higher as compared to the individual at the lower panel. So when we are looking at a recipient and donor, what we want is the best compatibility between the recipient and donor. And in case that's practically not possible because we always cannot find an identical donor, we need to understand how to control the immune response due to the mismatches. And for identifying the best match, we need to have a lot of good technologies to be able to do a good HLA match. So laboratories in immunogenetics field need to advance. We started with serology-based techniques and went on to Luminex-based techniques. And at the present moment, majority of the labs are doing the sequencing using next generation or third generation sequencing. So one laboratory should be able to expand or improve upon the newer technologies so that we can do a better selection of the donors. For example, at AIMS, we have almost all the technologies. So for the clinicians who are not well exposed to the laboratory parameters, I would just like to give you briefly that when we do a serology-based assay, we are looking at the lymphocytes of any individual. And then we are binding the lymphocytes with anti-HLA antibodies and trying to use some dyes, either fluorescent or non-fluorescent dyes, and observe under the microscope to detect what is the HLA antigen expressed on the surface of any particular cell. And this technology was actually defined by Sir Paul Terasaki, who has enormous contribution. And these are the HLA plates that they are named on Paul Terasaki's name. And we use these for, used to use this technology much earlier. But nowadays, this technology is only used for cross-matching, which is called the CDC cross-match. And people have moved on to molecular assays because we can get better resolution HLA types, and these are several HLA typing technologies which most immunogenetic labs which are maintaining state of the art would definitely be having the in use in the laboratories. So depending on the throughput and the resolution of the HLA typing that we are interested, any one of or two of these technologies would definitely be available in most immunogenetics lab. So currently you people must be all hearing about the Luminex based antibody screening. So this is a Luminex platform wherein we use this for HLA typing as well as for antibody monitoring. Using this platform, we can amplify, once we amplify the DNA and we know this HLA region which is amplified, we use the fluorescent conjugated beads, and these beads have the probes against the HLA antigens, and using the software, we are able to identify the HLA types. And the same technology is also used to do antibody assays, which we will talk about later. So this technology is also improved, and we have now about platforms where we can use 500 fluorescent conjugated beads, and we can do a deeper HLA typing, and a high resolution HLA typing can be done using these platforms. Further on, we can move on to sequencing, which is the standard Sanger sequencing, where we get the exact sequencing of the HLA regions, and we can identify where the recipient and donor differ. And we also moved on to the next generation or the third generation sequencing, where we can look at the complete HLA region and bit by bit and sequence it in hundreds of times, so that we are absolutely sure of every region, and then pool this data together to be able to get a complete HLA type, or full region is HLA typed, and we get a high resolution HLA typing. This can be done using multiple platforms. At All India Institute of Medical Sciences also, we have a couple of these platforms, and we can do a high throughput next generation sequencing based HLA typing. I can just highlight a few examples that using this technology, we studied the Naga tribal populations of India, and within a span of 6 to 7 months, we did just about few hundred samples, and we were able to identify this n number of novel genes. So these were new, which were not reported in the HLA literature, and we were able to find it. The point of my mentioning this is that we need to have these high resolution technologies, because if we do not have a high resolution HLA type, we will not be able to detect the good matching, as well as the antibody specificity, where we want that we need to identify the donor specific antibodies. So in today's world, usually the clinicians would think we have good immunosuppression, we've got HLA typing technologies. Do we just manage with immunosuppression? Do we need to look at the compatibility? But I think it's important for us to keep the HLA typing as a high resolution, and have, we can have better graft survival. We can have lesser, it will also lead to lesser frequency of de novo antibodies, and the toxicity due to immunosuppressive drugs will be lower. And once the patients are less sensitized due to better matching graft, even if the pediatric patients require another transplant later on in life, they will be less sensitized, and the outcomes of the grafts are going to be better. So in terms of organ transplant, if we look at the matching, if we have an identical HLA type, it's on the top panel, it's the best. Whereas if we keep on adding mismatches to this panel between the recipient and donor, if there is a complete mismatch of HLA type, that is the worst, and we would preferably avoid such a mismatch. But in case there is a mismatch, we should be able to predict what will be the impact on the immune system once we have a mismatch. So this is the data from collaborative transplant study in Europe, and this is many years ago, and I'm sure it's going to be right now also exactly the same. If you log in the website, that 0 mismatches for HLA leads to better graft survival when we compare to 6 mismatches, and it is applicable to kidney and all other solid organ transplants as well. So HLA matching do play an important role, and also is important because when we are predicting the antibodies, so many individuals who are on waiting list on transplant, many individuals who have undergone pregnancies and who have undergone previous transplantation do develop alloimmune responses and have antibodies. So we need to have good technologies to be able to identify what these antibodies are against. Are they against the donor organ that we are going to graft, or they are non-specific antibodies? And these antibodies are really bad. They could be harming the graft. If they are C1q binding, they could lead to ADCC, there could be opsonization, they could be activating the cells on the endothelial cells. And because of all of these phenomena, they could be impacting the allograft. So I'm just going to give you one case wherein if we do not have a good high resolution HLA matching, how we miss the antibody profile. For example, this is a recipient donor pair where we have not tested the HLA-C or the other HLA class 2 apart from DR. and we detect the single bead antigen assay, the antibodies to HLA-C or HLA-DRB1 in the lower region of the slide, we are not able to say whether these are the donor-specific antibodies or not, because we do not have the HLA-C profile of the recipient and the donor. No matter if we have a better profile, for example, this, where the HLA-C is typed at a high resolution and also the HLA-DR is typed at a high resolution, we see these two alleles in red color, we are able to say that yes, these antibodies are donor specific. So in absence of a high resolution HLA type or a good standards of HLA typing, our donor specific antibody predictions are not perfect. So we need to have better resolution HLA typing. So different HLA typing technologies are there, and the similar technology that we used initially for HLA typing is used for how we need to now identify, okay, we have done the HLA type and how we need to go forward to be able to detect whether the graft can be taken up. So we need to do a cross match, which is a serology based cross match. We just add recipient serum and add donor lymphocytes and estimate what is the reactivity towards the donor antigen. We can use flow cytometry based assays wherein we use fluorochrome based antibodies. However, in the flow cytometry based assays, we detect not only the complement fixing antibodies, but non-complement fixing antibodies also. Whereas in the serology based assay, we only detect the complement fixing antibodies. So there are advantages. We can use donor lymphocytes, and using the cell lysate, we can predict the donor antibodies. So this is not, with the time frame today you're not able to discuss all of this in detail. There will be other speakers talking about these assays. So there are different cross-matching technologies available, and every center needs to define their parameters when and how they are able to predict the alloreactivity using either the complement-dependent assay or the flow cytometry-based assay, or I know other speakers are going to talk on the virtual cross matches and how we predict the virtual cross matches. Idea is that we should be screening the serum of the recipients if they have any antibodies, and to what specificities do they have antibody. We can do that using flow cytometry. We can use that using single beads on the Luminex platform where they have beads. The whole idea behind this is the biology is that we should be able to detect which HLA specificities are crucial and not are a mismatch. So when we look at the HLA molecules, there could be some public epitopes which are common to most of the HLA profiles, but there could be some private epitopes. And there is also cross reactivity between one HLA specificity with others. So that can enhance the PRA levels. And because of this cross reactivity, one should keep in mind, for example, in this case, the allograft, if has an HLA-A2, that antibody could react with a panel of other HLA specificities, as you see in the lower panel in the pink. And these all can cumulatively enhance these PRA levels. So there are multiple such cross-reactive groups which the laboratory can help the clinicians that this is the calculated PRA. And based on this calculated PRA, one needs to estimate whether that recipient and donor are compatible or not. So we need to have technologies that are more sensitive and more specific. So we could have serology-based assays, ELISAs, flow, or Luminex-based assay. And there are standard guidelines published based on which, when and what investigations need to be done for solid organ transplantation. I would highlight a little bit on the virtual crossmatch. And there is a paper published, and I would encourage all of you to read this paper, which discusses, it's a debate on physical cross-matching versus a virtual cross-matching. In this era, what should we be doing? Should we give up the old, or is that good enough or not? And I would just like to mention a few things that I think we should not give up the old, because the gold standards are always helpful. However, there are advantages and disadvantages to both the technologies. For example, in virtual cross-match, we enhance the sensitivity, we shorten the time for the transplant, the cold ischemia time is reduced, and this is associated with the decreased wait list and mortality. However, there are disadvantages, and those disadvantages need to be managed. So we can, sometimes there could be reliant on current and correct donor typings for antibody identification, and we can do that using different modalities. There could be antibodies targeting denatured epitopes of the HLA molecule, and that can be salvaged. Prozone effect can lead to false negativity, and antibody against rare HLA alleles. So when I was talking to you about the novel HLA alleles that we identified in our population, sometimes we could miss if we do not have those beads on the single antigen bead panel, and we will be able to miss those novel alleles, and we can't predict properly. Similarly, physical crossmatch, there are advantages. We can reveal exactly the binding which is due to the actual cells when we are using. The accurate immunological risk can be assessed. Donor cells are best for doing this discrimination, and we remove the false positivity due to the solid phase assays. But there are disadvantages that we have to rely on the viability of the cells. We have to have pure cells, expression of the HLA profiles. Then sometimes the laboratory protocols need good handling, and we need to also handle the some auto reactivity, and there could be also backgrounds, but there are ways that we could salvage these disadvantages as well. So the laboratories need to have this in mind and support the transplant program and develop some algorithms that these pre-transplant patients, how do we work up, what are the assays which require for the high risk turn out to be high risk for the patients, what are the intermediate risk assays, and what are the low risk assays. And every laboratory need to have some kind of algorithm that for a living related donor program as compared to a deceased donor program, what investigations are we going to do initially and also in a post-transplant scenario. So some laboratories do have assays, multiple assays. And for example, this paper published from India from our center indicated that with a cutoff of MFI of 3000, they were able to predict a flow cytometry cross-match positivity. And similarly, with a cutoff of about 7000 MF5, the center was able to predict that this recipient donor might lead to a CDC cross-match positive. So having multiple assays and a correlation done in your own respective laboratory does help the transplant program to do it. So we also need to see when the antibodies are formed, what are the kind of antibodies, whether they are to class 1 or to class 2, and this publication, I'm giving some examples from kidney transplant, but the same thing applies to all other solid organ transplants. So this shows that the survival free, from the acute rejections is much better when there are no donor specific antibodies as compared to when there are preformed class 1 and class 2 antibodies. Further, these antibodies may develop and may clear, and some of the antibodies may stay on, and these are called persistent preformed antibodies. So the graft survivals are much lower. The antibody mediated free graft survivals are much lower when we have de novo DSA antibodies, and then compared to the persistent, and it's much better when there is no DSA or when the antibodies get cleared off. So moving on to the publication on lung transplant, this publication indicates that there could be antibodies which are transient and they come up early in the graft time, post-transplant period, and then they fade off, whereas there are certain antibodies which come up a little later and they stay on for longer time and have a poor impact on the graft survival. And these patients who have the donor-specific antibodies, and they have a much higher, ventilation time in the ICUs post-transplant, and their time in the ICUs is, as compared to the DSA negative recipients, is much higher. And these persistent antibodies also impact the survival. When the DSAs are transient, the survival is much better, but when the DSA is persistent or the DSA is negative, the survival is, persistent DSAs, the survival is inferior. So usually all of you who are performing the transplant would follow several desensitization protocols, and post desensitization protocols would like to see that the antibodies clear off. But the nature of antibodies actually impacts. For example, if the antibodies were both C1q binding and the C3T binding, they do not get cleared off, whereas the antibodies which are not C1q binding or only C1q and not C3D get cleared off. So the laboratory needs to provide information to the physicians and the surgeon and the transplant program. What is the nature of the antibodies that we have? Now, when the antibodies bind to the HLA specificities, I would just like to bring up few novel concepts that, so this is the epitope of the HLA antigen would bind to the paratopes on the antibodies. And this interaction is very important, and certain regions are very crucial. So it is not the complete HLA, but certain epitopes in the HLA molecule which are very crucial. So if we look at further in detail, that some epitopes which are identified and lead to immunogenicity, they are the functional epitopes, and they are the ones that can, so it is not the complete HLA molecule that could be immunogenic. There could be certain epitopes or certain regions, smaller regions within the MHC molecule that will lead to immunogenic response. And we need to, for example, in this slide, you see that the regions in the blue are the immunogenic epitopes, and the regions in the red are the crucial amino acids. So presence of three of these in the top panel, in the middle row and the top, you can see that both the crucial amino acids in red and the immunogenic epitope would lead to a cross-match positivity or alloreactivity, whereas absence of any of these will not lead to immunogenicity. So one needs to understand which epitopes are immunogenic. And this is done using several softwares called HLA Matchmaker. So looking at the HLA sequences, these applets and the epitopes, the load from the donor can be predicted, and one can predict the outcome of the transplant. And for example, if you see in this slide that there are self epitopes which are in blue, there are the unacceptable epitopes in red, and there are the acceptable epitopes which are acceptable mismatches. So if we have a self epitope and no mismatching with the self epitope, then there would be no positivity of the cross match. Whereas in the scenarios where there is an unacceptable epitope, the cross match would be positive. So we can predict these antigenic or immunogenic epitopes and identify the acceptable versus unacceptable mismatch, which has led to the concept of virtual cross-matching. So for the interest of time, I will move forward and just highlight a few points that in cardiothoracic transplantation, the HLA-DQ epitopes have been identified as very, very crucial. And there are studies that have identified that if we look at the graft survival, the cumulative de novo DSA-free survivals, this survival is much better when we have no mismatch, or if we have a DQ mismatch, then it is reduced. If we have these risk epitope mismatches predicted by the software, the survival goes even worse. And if these risk epitopes and DQA epitopes are present, then the survival is even the worst. So we need to identify which are the immunogenic epitopes. This is from the cardiothoracic transplant program, and very clearly indicating that the risk epitopes need to be identified. And there is a recent development in the last few years wherein the algorithms have been developed, which is called PERCH. And this is called predicted indirectly recognizable HLA epitopes. And this takes care of, the indirect allo recognition, which I talked in the very beginning, that the antigen presentation can be in two modes in the organ transplantation. So the indirect antigen presentation is predicted using these softwares that are developed. And these predictions can also indicate us whether there will be a cytotoxic T cell response or there will be HLA antibody responses in the post-transplant scenario. For example, if we look into this slide, the first top panel indicates the HLA typing between a patient and a donor. And the red boxes, red outline boxes indicate the mismatches. So the softwares like PERCH can predict what are the kind of peptides that these mismatches will lead to. And when those peptides are presented, they will be identified by the graft. And this PERCH algorithm also helps us to identify what is the probability of any individual having the PERCH mismatch. For example, in this graph, you see the blue bars indicate that these are the PERCH scores of a recipient donor pair. And if it is lower, there is a high probability, about 60%, this 10%, the green box and the yellow box, that they would be able to find a donor. Whereas if the PERCH score predicted by the software is higher, there is a very, very low probability, only about 1%, that this recipient might find a good donor allocated from the programs. So these HLA predictions have helped in many programs, and this is a paper in which one of our panelists, Dr. Patel, is also part of, that they identified the DQ mismatches and have correlated that how these impact the heart transplant recipients. And using these predictions for the HLA DQ de novo antibodies, they are able to predict that the patient survival is inferior when they are the DQ de novo DSA, and it matches with the the applet scores that we estimate using the HLA matchmaker, as well as the perch epitope mismatch scores when we use the perch software, and we are able to predict how much of the alloreactivity this organ might generate, and then we should be allocating these organs or not. And the freedom from the de novo DSA has been identified using these programs of HLA matchmaker as well as perch, and all of these predict that that when we are able to predict a better and allocate the organ according to these predictions, the the patients are free of the antibody mediated rejection. So I would just introduce one more concept
Sorry, I think we are running short of time because we have 4 more speakers.
So the last concept I would like to just mention is that now we can also do the mismatching predictions and estimate the statistical potential, electrostatic potential of the HLA molecules. These modalities are now used that we can predict the structure, and using the electrostatic potential we can identify what is the mismatch score and what will be the outcome. And these matches with the predictions match with the HLA matchmaker as well as the PERS scores. And some of the new concepts have come up apart from the classical HLA, that there is also a role of non-classical HLA molecules, which are also located on chromosome 6. They play a role in organ transplant, and especially in heart transplant and cardiac transplant. There are several publications indicating that we can also use these HLA-G expressions on the allograft and the biopsies and estimate how the benefit that we can get with the HLA-G expression, as well as the soluble levels in the serum can be predicting the infections post-transplant, the malignancies post-transplant, and the survival post-transplant. So these are some of the new concepts that we need to keep in mind when we are having a transplant program for cardiothoracic organs, as well as people have started estimating the donor-specific antibodies using the graft tissues. So this is the intragraft DSA assessments also help us in predicting how the rejection can be monitored. I would wind up here and once again thank the Indian Society for Heart and Lung Transplantation for organizing this wonderful Congress and giving me an opportunity to highlight a few points that we need to keep in mind for a transplant program. Thank you so much for your patient hearing.
Thank you, Dr. Uma, for the wonderful presentation. It would be of great help in the future discussions. The next speaker is Dr. Dolly Daniel, Professor of Transfusion Medicine and Hematology in CMC Vellore, and she will be talking on important issues like panel reactive antibodies and CDC crossmatch. So over to you, Dr. Dolly.
Thank you, sir. Thank you to Dr. Abhikma and the team for inviting me to be part of this meeting. In 1969, when the complement-dependent cytotoxicity crossmatch was described by Patel and Terasaki, I think we must have all reached a new high, a point when we thought every bit of diversity in the area of transplantation could be actually embraced. But I think as time went on and tools to define HLA and anti-HLA antibodies became more available, as Dr. Uma had explained, an antigen of the HLA system became an allele, and now with high resolution typing, you can see that what was first an antigen has now become an allele. And therefore, the number of antigens and alleles at any given locus are just numerous. The tools that we had, sorry, my slides are just moving by themselves. The tools that we have to actually detect anti-HLA antibodies have also evolved over time. And from the basic complement-dependent cytotoxicity test, which was described earlier, we now have ELISAs, we have flow cytometric cross matches, and we have the very sensitive Luminex-based platform as well. We all know that HLA matching is good and anti-HLA antibodies are bad. And we have tools to actually detect both of these in much greater detail than before. And in a sense, we can say that we're actually spoiled for choice. So what then is the problem and why the controversy in what we use? The problem is that the techniques are plentiful, sensitivity varies, but the clinical impact of what we actually detect is also variable. We can have interference with non-HLA antibodies in some of our assays, and therefore the clinical impact of what we actually find can be extremely varied. I have a lot of respect for Dr. Franz Claus, who many years ago, who said that in the midst of all this technology, HLA antibody testing should be done to facilitate a safe transplant and not to actually prevent what could be a safe transplantation. And therefore, it is against this background that we need to look at the tests that we offer. Let me just illustrate to you what I mean. This is a very elegant study which was published in the NEJM a few years ago. And here you can see, this is in the renal transplant group. So they've divided these patients into low risk based on a mild DSA and a high risk based on a high DSA. And you can see here that the top rows are those who have been transplanted. And just look at the bottom rows where patients were never transplanted. And you can see that there's a huge survival benefit which has been achieved by patients who actually got transplanted, even in the high-risk group, 71% versus 43%. And therefore, we have to look at our data and our tests carefully before we decide to deny or accept somebody for transplantation. In the lung scenario, in the lung transplant scenario, this is a study, a retrospective single-center study, but which has looked at more than 700 patients. And they've come to the conclusion that in the high-risk group, that is those who are broadly alloimmunized, there were longer waiting times and more people tended to die on the wait list itself. But on the other hand, in heart transplant, they found that survival on the waiting list actually got better, and they attributed to better improvements in the management of heart failure therapy. So truly, the old order had changed. And today we have a wide variety of tools, and we need to look at it against this, which brings me to my topic, preoperative complement-dependent cytotoxicity and panel reactive antibody testing in the context of what is happening in India at the moment. So before I go into what's happening here, let me just introduce you to the different platforms and the different formats we have for antibody testing. As Dr. Umar mentioned, the platforms we have vary from the complement-dependent cytotoxicity, which is the least sensitive, to ELISA, flow cytometry, and finally the Luminex. And this is in increasing order of sensitivity, with the Luminex being the most sensitive. Now we have different formats which are put up here, which is a screen, cross-match, virtual cross-match, and a panel reactive antibody. So what is a screen? A screen is basically when you do a test and it just tells you, are there antibodies or not? So are there anti-HLA antibodies, class 1, class 2 or not? A panel reactive antibody talks about the breadth of sensitization, and a cross-match actually tells you whether you have antibodies against one specific donor. And this can be physical, where you use donor material, or it could be virtual, as in the single antigen bead assay. So looking at the CDC, which is the oldest of tests to detect antibody, it's very important to realize that the main limitation of this is its sensitivity, and therefore it should be enhanced. So we enhance it with extended incubations, adding antiglobulin reagent, and it also detects a mixture of both IgG and IgM antibodies. So we use DTT to decide whether it's an IgG or IgM, and I'll come to that shortly. Also, it only detects complement fixing antibodies and can detect non-HLA antibodies as well. The PRA, on the other hand, just tells you how widely sensitized is this individual, and any platform can be used. You can use a complement dependent, you can use the Luminex based. But right now, the PRA has gone out of vogue, and what is important is a calculated PRA, and that I'll come to shortly. So this is just a picture of the complement-dependent cytotoxicity many of us are familiar with. The bright things here are the live cells. So we grade the number of dead cells. And these dead cells look totally bland. Now, if a cross-match is positive, we need to use chemicals like DTT, which actually break the disulfide bond of the IgM molecule. And if a cross-match becomes negative after the serum is treated with DTT, then you know that it is an IgM antibody. If it persists, then it is an IgG antibody. Now, if we identify an IgM antibody, it is very important in a donor-recipient cross-match. It is very important to give it time and retest it 2 to 4 weeks later to make sure that there's no class switch, because it could become an IgG, which is clinically significant. Let me just give you the example of a 24-year-old lady who came for a transplant workup. Now, she was matched as a haplomatch with the mother. And if you look at this, there was a weak positivity in both the auto cross-match as well as the donor-recipient cross-match in quite a few dilutions. Now, once the serum was treated with DTT, you can see that this was negative. Okay? And so this was suggestive of an IgM antibody. A flow cross-match was done and the T and the B cell was negative, repeated after 2 weeks, and we suggested that the cross-match is repeated after 2 weeks. 2 weeks later, exactly the same, but the flow cross-match was positive and the Luminex single antigen bead assay showed an antibody. So this is very important to remember that when you find somebody positive on a screen, even if we do not detect IgG antibodies, that we add on sensitive platforms to make sure that there is no underlying small amount of an IgG antibody lurking. Now, the calculated PRA that I talked about, the calculated PRA is basically going backwards. So when we find an antibody specificity, we then look at how frequent that particular antigen to which the antibody is directed is present among the donors from that particular ethnic population. And therefore, it is an ethnically weighted reference database. And a calculated PRA of more than 50% is considered highly significant and is often considered an indication for desensitization. Now, using this, we have to also make sure that we label what are called unacceptable antigens. And therefore, it is very important that when an antibody is identified, that we know what high resolution allele it is identified against, and therefore it is advisable that our patients also have a high resolution typing. If we put in a low resolution antigen, then we find that the CPRA just massively increases and it becomes very difficult to subsequently look for a donor as well. So how does this impact on lung transplant? So this is a study where they looked at more than 10,000 lung transplants, and they divided patients with less than 25% and more than 25% PRA. And you can see that there's a split in the curve and that there is a significant, significant difference between those with outcomes of those with greater than 25 and less than 25. But interestingly, they then split it out time-wise. And the ones which are done between 87 and 97, there was a significant difference. But as you came to more recent years, this really bridged the gap and there was no significant difference. Again, this is another study where they did lung transplants in patients who had donor-specific antibodies, but they made sure that they were CDC negative. And not unexpectedly, they found that the number of antibody-mediated rejections in these patients was significantly higher. But on the other hand, despite the DSA, the chronic lung allograft dysfunction was really not different in both these groups with and without DSA. Now moving on to hearts. This is a really nice study done in the pediatric group where they looked at 4 groups: non-sensitized, sensitized with DSA, and sensitized with DSA and a CDC positivity. I'll go straight to this. And here they look at antibody-mediated rejection and cellular rejection. And these two are the ones where you actually have a problem. And you can clearly see that the number of patients with antibody-mediated rejection, cellular rejection are much higher than the other two groups where the CDC was negative. And this green line on top is a cumulative number of the number of AMR and ACR episodes, and the green line stands way above those without a CDC positivity. There's a very nice paper. It's a statement from the American Heart Association along with the International Society for Heart and Lung Transplant. And here they have endorsed the use of the calculated PRA. They've also talked about more than 20 or 25% of PRA being significant. And they've acknowledged that sensitization is associated with post-transplant mortality, and therefore it is good to avoid it. Interestingly, they've also talked about the Luminex-based single antigen bead assay, focusing on the importance of complement fixation in the antibodies which have been identified. They go on to say that transplanting across a positive CDC is really not typically recommended, and that if you have a CPRA greater than 50%, the patient should probably be desensitized. The consensus guidelines that Dr. Uma mentioned are also along the same lines, and they focus on the importance of risk stratification in thoracic organ transplants and the necessity to avoid CDC positive transplants. And if we could minimize the risk of sensitization by blood administration of blood products, then it's a good thing to do. The importance of having pre-transplant material stored, both DNA as well as pre-transplant sera, is emphasized in the consensus guidelines because it serves as a referral subsequently for anybody who needs to do the test. Now, so what happens in India? In India, we have heart transplantation for end-stage renal failure, a single center experience of 257 patients. This is one of the largest series published a short while ago. And here, the reference to PRA is in the context of pre-transplant evaluation where they all had one. But subsequently, there is no analysis of whether patients actually responded differently or whether outcomes were different. But post-transplant, if there is a problem, they all have single antigen bead assays. There are two editorials focusing on the need for better immunological monitoring in India by Dr. Mishra and by Dr. Balakrishnan. So in a country where we've gone this far, why is it that transplant immunology testing is such a challenge? And I think it is a challenge because of the wide disparity in our population and the big divide between the poor and the rich. And cost goes up with testing. And you can see that a CDC, which costs about $30 US or 1,500 to 3,000 bucks, and a single antigen bead costs more than 20,000 bucks. But there is a beautiful study from Sweden where they have looked at the effect of kidney transplant, focusing not just on graft outcome, but about how many people actually went back to work after renal transplant. So my friends, is a huge elephant in the room. If in a developed world like Sweden, they are looking to see how many of them go back, how much more important is it in a developing resource-constrained country? Because for a patient who comes scraping the bottom of the barrel for a transplant, this is his or her only chance. And therefore, it is imperative that we give them the best chance of longevity of graft. We need to understand transplantation is here to stay, and how can we make it safer is a question that we ask. And if we give them greater longevity, in the long run, it is financial savings. So therefore, it's important to remember that whether or not there is a commercial opportunity, there's a huge human opportunity in resource-constrained settings. And therefore, let me just tell you what we do in renal transplant and disease. It's very similar to what we do, what we need to do in thoracic organ. And definitely, it's not a wild goose chase. We need to choose what we need to do. The first thing is to stratify our patients immunologically into high risk or low risk. And the questions we ask are a careful history. Is it a second transplant? Is there a historical CDC positivity? And in heart, ventricular-assisted devices very often are a high risk for sensitization. Somebody who's had a Luminex cross-match positivity. And now we know that even unsensitized males have a big problem with the antibodies. So this is an algorithm we have for a patient going on a wait list for a diseased organ. We first assess risk, history, and historic cross-match positivity. We perform a high-resolution HLA typing by the NGS. And if this is a second transplant, we try our best to get a high-resolution HLA typing of the previous donor. A single antigen bead is done every 3 to 6 months or 2 to 4 weeks after any immunizing effect. We have not been able to put complement binding into the SAB because of costs. And we have not been able to put a CPRA inside because we do not have that data. But based on this, we stratify them into different levels of immunological risk. When there is a call for a deceased donor organ, then the next set of tests that we do takes 4 to 5 hours. We do a CDC with and without DTT. We do a flow cross-match, and usually 3 to 4 possible recipients are tested together in case, particularly when the first 2 are high risk. We HLA type the donor with an intermediate level resolution test, and we find that we can actually project a high resolution from it. And if the single antigen bead assay has been done more than 3 months earlier, then we go ahead and do it. We look at unacceptable antigens, which we have marked out, particularly if the patient has had a previous CDC positivity or this is a second transplant. And the single antigen bead assay is interpreted in that context with the donor HLA typing. So this is to re-emphasize on if it's a second transplant. Now, there's a lot of focus on the virtual cross-match and how good it can be, but just a word of caution, this is something that has been published from our center very recently about the physical cross-match versus the virtual and whether it can go as a singular tool. These two graphs just show you two things. The one on the left shows you that 50% of class 1 and class 2 alleles were not represented on the single antigen bead panels of 2 different vendors. The second graph shows you that 85% of our patients, in 85% of our patients, all alleles were not represented. So therefore, to go with a virtual cross-match as a singular tool is extremely dangerous. We need to allow flexibility so that within the Indian context and constraints, we are able to come up with the best possible combination, making sure that we plug the right hole. And that is the most important question. Prevention is better than cure. So if patients need transfusions, give them leukodepleted products, they stand a slightly lesser chance of alloimmunization. This has always been a challenge, transplant immunology, introducing it because it's considered high-end, lack of Indian data, and harmonization can be difficult. But I think it is very important that accessibility to safe patient care is an issue in justice for our patients. And more than equality, what we need is to be able to give them equity in the way we treat them. So I think we have come so far with our organ transplantation, be it renal, be it thoracic. But it is important to listen to those voices external that sometimes we don't always hear, to see how we can actually make our transplant graft longevity even better for the patient community that we share. There are teams which work behind it. Many people have gone through the labs, and it is important that we carry our team together to make this happen. And let me end with one of my favorite quotes by Vince Lombardi. I think being in a developing country, in a resource-constrained setting, perfection may not always be attainable. But friends, I truly believe that if we chase perfection, we will definitely catch excellence. Thank you.
Thank you, Dr. Dolly, for your wonderful presentation. I mean, yeah, I think touch upon all the important topics or problems also we encounter in India. So our next speaker is Dr. Evan P. Kranstoff, Senior Transplant Cardiologist at Cedar Sinai, California. A very long experience he has in heart transplant practices, donor organ allocation. So over to you, Dr. Evan.
Thank you very much. It's really a great honor for me to be here today and to present to you. And the topic I'm going to speak on is the importance of virtual cross-match and HLA immunogenetics for thoracic organ transplant. And I'm really going to focus on heart transplant because that's what we do. A little bit of flavor from the US experience, And I think my comments are going to overlap somewhat from the comments of Dr. Kanga and Dr. Daniel. But I think there's a little bit of insight we can kind of shed on this topic, and maybe propose some directions for future collaboration, even to just sort of overall implement some of the strategies we've utilized to assess this area. My specific areas I'm going to talk about, I'm going to present some data highlighting the effect of allosensitization on heart transplant outcomes before and after transplant, discuss some of the strengths and limitations of the virtual crossmatch while trying not to overall duplicate the really excellent points that have already been made. And I think importantly, describe the calculated panel reactive antibody as a measure of allosensitization, as well as highlight some of the work we've done in that area in the US, showing that our CPRA metric had quite a bit of room for improvement. And I think this is something that could be implemented in India as well. So just first turning to some of the data that looks at the role of allosensitization in heart transplantation. So this is some work that we did a few years ago. Basically, we used the Organ Procurement and Transplantation Network US database that has information on outcomes of heart transplant candidates and transplant recipients. We looked at a cohort of patients between 2006 and 2013. The issue is in the United States, only a small portion, around 10% of candidates, actually have their unacceptable HLA antigens recorded in the database. And that's for a variety of reasons, but most importantly, that the unacceptables don't achieve anything for the candidate in the United States, as opposed to kidney transplant in the United States, where patients receive allocation priority based on their level of unacceptable antigens as measured by the CPRA. Given the fact that that doesn't get you anything in the US, very few programs actually enter that data. But we did, again, find about 10% of candidates that did have their unacceptable HLA antigens that were entered in the database. And so we used this as a basis to do a study. Basically, for each candidate, we calculated their CPRA while they were waiting for a transplant. And then we used that to look at outcomes on the waiting list. What you can see here on the x-axis is that we've divided the candidates into 5 different groups based on their level of CPRA, grouped by sort of different 20% increases in their CPRA. And then on the y-axis there, we have the percentage of patients in that group that had that outcome. So we can see as we go to progressively higher levels of CPRA, what we see is that there are more patients waiting for a transplant, more patients removed from the transplant list for getting too sick, more patients that died, and less patients that get transplanted. So as we would expect, CPRA is a very strong predictor of outcomes while patients are waiting. And something that I thought was important that we found from this study is we were able to assess in each of these groups how many patients were listed for a prospective physical crossmatch as opposed to use of the virtual crossmatch. And what we found is as the CPRA got higher, more and more patients were requested to have a physical crossmatch. So although the US, we do have a very broad experience using the virtual crossmatch, as you can see from this data, there's still a very strong utilization of the physical crossmatch, especially in sensitized patients and with progressive increase as the level of sensitization increases. We then also looked at outcomes after transplant, and what you can see here, the different colors represent the different levels of CPRA, with the x-axis being the time post-transplant and the y-axis being survival. So the green group there is control patients who had no listed unacceptable HLA antibodies. The yellow group is the 0 to 20% CPRA, and the various sort of orange to red colors represent increasing CPRA. And what we can see here is that in the sensitized candidates, they have a lower freedom from death after transplant, and this is looking up to about 3 years. But of course, it's not a linear relationship because of all the different reasons that Dr. Kanga and Dr. Daniel have highlighted, that there's a variety of different ways to look at the effects of sensitization beyond just the level of CPRA, specifically including whether the antibodies are C1Q, the level of mismatching between the recipient and the donor, at the serological level, at the molecular level. So, from a CPRA standpoint, it's not a linear relationship, but we know certainly that there is a strong effect of sensitization on outcomes post-transplant, with these sensitized patients having a higher risk of death that still remains fairly unmitigated in this population. I'm going to spend a couple slides just talking about our experience with prospective cross-matching versus virtual cross-matching in the United States. And once again, I echo the sentiments already made by Dr. Kanga and Dr. Daniel. But as I mentioned, we generally speaking use virtual cross-match for most patients, and then sort of in patients with intermediate levels of sensitization, let's say a CPRA of 50% or so, it's very frequent that we'll do a virtual cross-match and a prospective cross-match. And then in patients that are highly sensitized, CPRA 80%, we're certainly going to use a physical cross-match to get a better read on the donor at the time of the offer. So just kind of in terms of basics, for the prospective cross-match, we're talking about donor cells and recipient serum put together with a readout of using cytotoxicity in terms of cells lysed or with flow cytometry. Obviously, cytotoxicity assays are going to offer a higher level of specificity, but flow cytometry is going to offer a better level of sensitivity. And of course, with the virtual cross-match, we're talking about Luminex single antigen beads for the most part, combined with recipient serum, and the readout intensity is the intensity of fluorescence. This slide has actually already been shown in the talk so far. I think this is actually one of the nicest reviews, certainly in terms of our figures that explain the prospective cross-match versus the virtual cross-match. And I certainly won't dwell on this since it's already been covered several times this morning. I think this is a very nice paper by Anat Tambor at Northwestern that looks at some of the data behind the use of the virtual cross-match as compared to the physical cross-matching. And basically in their study, which now is about 12 years old, they found that the sensitivity of the virtual cross-match was around 86% with a specificity of 97%. When you go through this and you look at some of the data that I have highlighted in this figure, what you actually see is that in their data, actually the specificity of the virtual cross-match decreased at higher levels of PRA. And now this is, I think, an important point, but not only in our experience does the specificity decrease, meaning that you may detect antibodies that may not necessarily lead to cell killing or rejection. But in addition, I think there is certainly data that sensitivity is also decreased in terms of proserning and other effects like that. This is, I think, some of the best data from the United States looking at the effect of the virtual cross-match on waiting time. In the US, the reason why virtual cross-match is very helpful is because it allows us to avoid having to bank serum at the OPO labs. As a result, we're able to better and more quickly evaluate potential organ offers. And also we're able to evaluate offers that are made outside of our local region, maybe coming from several states away. This allows us to proceed to transplant quicker, to have more donors available. And this is a nice paper from our group from about 10 years ago that just shows that using virtual cross-match, the time to transplant was significantly decreased. You can see there from a median of 36.5 to 27.5 with a huge difference in the maximum time spent waiting for transplant. And this was not accomplished with any increase or adverse events. In fact, we didn't find that there was any difference in survival or rejection for patients using the virtual cross-match. So again, we find this to be a very helpful procedure. We use it for basically the vast majority of patients. Again, in patients who are sort of intermediate to high sensitized, we will use a virtual cross-match plus a physical cross-match if we think there's certainly a risk that the donor is going to lead to cell killing. And in patients that are highly sensitized, then we're certainly going to be using it in that situation. So just a couple slides about the strengths and weaknesses. Again, I'm going to go through this relatively quickly since we've covered a lot of this. But in our program, use of virtual cross-match expands the donor pool by decreasing the need for a physical cross-match, thereby increasing the number of centers, increasing sort of the region with which we can get offers from, and the effect of that is decreasing waiting time. On the positives, it does cover a wide range of HLA antigens. And in general, it has very excellent specificity, especially when you're considering all comers, that is a lot of patients who don't have any antibodies at all at baseline. Limitations, of course, as we've discussed, that for patients that are highly sensitized, the prozoning phenomenon is well documented, and studies to avoid this, such as dilution studies, can be very helpful. I think another limitation is that sensitized patients may have dynamic changes in their antibodies while they're waiting for transplant. Things can change, between 3 months ago and now. So having the most updated antibody profile is important. I think, especially in terms of the virtual cross-match, the ideal threshold for positivity is unclear. And of course, a lot of data has been published regarding use of accessory studies that can help with that. So for example, titration studies, dilution studies, C1Q studies, those all can be helpful to determine which are the significant antigens. And then I think as Dr. Daniel's work has really nicely published, the HLA allele representation on the beads is highly biased. And that unfortunately leads to the inability to really use virtual cross-match in all populations. And that's something that certainly needs to be addressed by the companies that produce the beads. Now I want to switch gears a little bit and talk about an area where we've done some work, which I think is highly relevant, and that is the calculated panel reactive antibody. So I know that this audience is very familiar with this concept, but I just want to talk a little bit about it. So what is the CPRA? Well, the issue is when you have the Luminex single antigen, it's difficult to use that to determine how many donors the recipient is incompatible with. So the calculated panel reactive antibody is a way to use the unacceptable antigens that you identify with a Luminex single antigen or a similar screening test, use those antigens as input to determine how much of your donor population is going to be incompatible with the recipient with that set of antigens. So said in another way, it's a mathematical method to quantitate the percentage of the donor population incompatible with a transplant candidate given the set of unacceptable HLA antigens. And what I think probably everyone is familiar with is that it utilizes a reference panel of the HLA haplotypes. And these reference panels are based on the population's ethnicity. That is, in the United States, for example, as I'll show you in the next slide, we use a reference panel that has Caucasian donors, African-American donors, Hispanic, Latino donors, and Asian Pacific Islander donors, because that's what represents the majority of our donor population, although not complete totality of our donor population. This is a figure from a recent paper that really kind of gets into the nitty-gritty of how CPRA is calculated. And I do want to spend a little bit of time with this because I think it's important. So basically, in the top left there, what you do is you have a set of unacceptable HLA antigens. For example, in this patient, it would be DR2 and DQ6. And then what you do is for each ethnicity, you look up in the haplotype reference panel what the frequency of those antigens are in those various donor populations. And it's not just a single frequency, it's going to be each antigen and its combination. So for example, for DR2 DQ6, you would have each antigen individually as well as that combination of antigens. And the level of complexity increases with the number of antigens. For example, with the number of loci. And for example, if you have 5 loci, you're going to have quite a number of different combinations that have to be looked up. So from those haplotype reference panels for each ethnicity, you get an ethnic specific CPRA using that calculation. and then you multiply each of those towards the weight for that ethnicity in the population. And as you can see in this figure, those are the different ethnic weights that are used in the United States based on the national frequency of those donors in the population, with Caucasian being about 69%, African American being about 15%, Hispanic being about 14%, and Asian Pacific Islander being about 2.3%. So that's all well and good, except that it's not quite accurate. And there's a few reasons for that. And this recent paper that we put together really highlights some of the inaccuracies in the CPRA calculation. And what we found was that there were two main factors that were responsible for the inaccuracies. And we determined those inaccuracies by using a reference panel derived from stem cell transplantation and compare that to the reference panel that's been derived from solid organ transplant donors. So one level of inaccuracy derives from the fact that the solid organ transplant reference panel is much, much smaller than what's available from stem cell transplantation. This stem cell transplantation data set has around 6 million volunteer donors in it, as compared, as you can see there, to the drastically smaller solid organ transplant panel. What we also identified is that when the haplotype frequencies for the solid organ transplant panel were derived more than 10 years ago in the United States, what it turns out is that there were some mathematical errors that were made, probably because of limitations in computational accuracy at that time. And so what we found was that when we looked at patients, especially in highly sensitized groups with CPRA over 90%, we found that the NMDP CPRA, that is a CPRA metric that was calculated using stem cell donors, was more accurate than that derived from the UNOS panel. And I think, again, highlighting the work of Dr. Dano and what she mentioned is, you may say, well, these look like pretty small changes, but when it comes down to it, it really does affect the equity of transplantation, because these CPRA calculations are important for determining patients' priority for transplant on the kidney transplant list. Again, they're not used in heart transplantation at this time for allocation, but they certainly are used in kidney transplant. And so what we found is that there were kidney transplant patients that were getting too much prioritization based on their CPRA, and some patients that were getting not enough CPRA benefit based on this. So basically, the sort of final word from this analysis is that HLA typing derived from volunteer stem cell donors improve the accuracy of CPRA values for highly sensitized kidney transplant candidates. And I'll add that the United Network for Organ Sharing is actually moving to a system where they calculate CPRA based on stem cell donors. That's also beneficial because it will also allow us to have a full CPRA, if you will, and that an important limitation of CPRA in the United States at this time is that it uses input HLA antigens from HLA-A, HLA-C, HLA-B, HLA-DR, HLA-DQ. B, it does not use HLA-DQA, nor does it use HLA-DP. But using stem cell donors, because that typing is available, we'll be able to make a full CPRA that includes all these loci. And that will certainly improve equity, further improve equity for patients who are sensitized. Now, something I'll add, and I think we'd be very interested in collaborating with the groups that are represented in this meeting, that in this US data set, because it is so large, there is HLA typing derived from volunteer stem cell donors for 21 different ethnic groups, which includes South Asian Indian. And there's pretty good representation there. So I think a potential collaborative project that we'd love to help with using the methods that we've already developed for the project which I presented is I think we could certainly help develop a CPRA metric that would be helpful just in terms of broad risk stratification of sensitized patients. And of course, there's going to be dramatic population substructure of HLA in the South Asian Indian population, depending on geography and many other factors. But certainly something is always a good place to start, and it can be further refined over time. I'd really like to thank everyone for the opportunity to talk. It's really been a pleasure and an honor to be with you today. And I'd be very happy as part of the panel discussion or through comments to answer any questions that anyone has.
Dr. Kransdorf. Wonderful presentation. Nice message given. And now we have a stalwart now talking, Dr. Jignesh Patel, who is co-director of the Heart Transplant Program, Seda Sinai Heart Institute, LA. Welcome, Dr. Jignesh. Your talk.
Dr. Ravi, thank you so much for this kind invitation. It's always a pleasure being with the Indian Society of Heart and Lung Transplantation and seeing it grow by the year. I'm just disappointed that we couldn't be physically together this year, but hopefully this will change next year. I'm today going to talk about donor-specific antibodies and how they affect cardiac transplantation and some newer advances relating this in this field. These are my disclosures here. You already saw this graph from Dr. Kransdorf's work showing the impact of sensitization on patients awaiting heart transplantation. The higher the rate of sensitization, the lower the rate of transplantation and the higher waitlist mortality. And there are a number of risk factors for sensitization, including multiple blood transfusions, recurrent infections, prior allograft transplants, prior cardiac surgeries using homografts, gender, particularly females who are multiparous, tend to get sensitized from paternal antigens. Ethnicity, African Americans, for example, in the United States tend to have a much higher level of sensitization than other groups. And increasingly what we're seeing are the sensitizing effects of durable ventricular assist devices. And this is as almost half the number of patients getting transplanted in the United States are bridged with a ventricular assist device. This has been a significant issue. But as we've heard this morning, not all antibodies may be the same. We know that up to 11% of patients undergoing heart transplant actually get transplanted across a donor-specific antibody. But after heart transplant, up to 30% of patients actually develop de novo donor-specific antibodies. And we've heard that really the development of antibodies against the allograft is associated with adverse outcomes. But not all donor-specific antibodies are the same. These DSAs can occur early after transplant, or they can occur late after transplant. They may be transient or persistent, or they may be at a lower level, higher level, or even complement fixing. And these antibodies may be HLA class 1 or class 2. And I'll provide some data as to whether showing that not all antibodies are the same, even if we think that they may be potentially harmful using the higher sensitivity assays that we've heard about this morning. And of course, once you have a donor-specific antibody, this leads to an increased risk of rejection or chronically the development of allograft vasculopathy. And as Dr. Kransdorf has shown, it's associated with a higher mortality. And this has been also demonstrated in earlier studies as well. And our group has certainly shown that patients who are transplanted across donor-specific antibodies, well, they actually do quite well if the flow cross-match at the time of transplant is negative. And their outcomes is actually the same as those that were transplanted without donor-specific antibodies. However, if we transplant patients with donor-specific antibodies, but these patients have a positive flow cross-match, then they do have a higher risk of antibody-mediated rejection. Conversely, when we look at the development of de novo DSA, we see that these patients actually have the worst outcomes with regard to antibody-mediated rejection, mixed rejection, or even graft survival. And this is much worse than patients who really have no antibodies at the time of transplant, or even have been transplanted across DSA at the time of transplantation, so that and these DSAs are persistent following transplant. Some recent data from Canada suggest that if you have de novo DSA, not all of these may be deleterious. Only those that are persistent following the development of de novo DSA after transplant are associated with a higher mortality. In this cohort of almost 200 patients, they found that 42 patients developed de novo DSA, and then they split this group into those that had persistent DSA at 6 weeks after the first test, or those that had a negative DSA 6 weeks after the initial positive Luminex assay. And they had 15 patients in the transient group and 27 patients in the persistent group. And what they found was that those patients who had persistent de novo DSA had a 4-fold hazard ratio for mortality that was statistically significant. And we've looked at some data in our group with regard to crossing donor-specific antibody at the time of transplant and seeing how these patients do. We have a very high number of patients that are referred to us with allo antibodies. And for many of these patients, we have been able to undergo transplantation with crossing antibodies at low or moderate levels. And as you can see here, the MFIs of the antibodies range in the 5,000 or so median fluorescence intensities. And you can see that these patients actually, as time goes on post-transplant, the MFI levels of these antibodies actually wane. However, in about a third of the patients, there is an amnestic response. What do we mean by this response? These patients actually have a rise in their antibody titer, not titer, but the antibody level following transplantation. And this rise in their antibodies, which usually occurs between 1 and 3 months after transplant, subsequently subsides in most cases, although not all cases. And is there a consequence to this? Well, patients where we did cross DSA certainly had an increased risk of either any treated rejection or antibody-mediated rejection. Most of these episodes occurred very early after transplant, within the first month or two. And this was certainly greater than those, the rejection rate was greater than those for whom we did not cross any donor-specific antibodies. However, when we looked at 1-year freedom from cardiac allograft vasculopathy or even graft dysfunction, we really saw no difference between those patients that we crossed the low or moderate level DSA and those that were not sensitized at the time of transplant. And therefore, we concluded that accepting low-level DSA at the time of transplant appears feasible and could certainly broaden the donor pool and shorten the wait time to transplantation. Well, what about antibodies that develop early after transplant versus late after transplant? Well, there's increasing data to suggest that those patients that develop antibodies late after transplant actually do much worse than those patients who develop antibodies early following transplant. This is data from Clerkin's group showing that the freedom from death or retransplant following the development of antibody-mediated rejection late after transplant was significantly lower than for patients who developed AMR early following transplant. And Dr. Coutances, who will be speaking after me, has also shown that the survival after late antibody-mediated rejection is very poor, at as low as 50% at 1 year. So certainly there appears to be a difference between whether you have antibodies, deleterious antibodies early versus late after transplant. And Dr. Lupi's group also has shown data looking at late failing allografts. These are all patients who underwent redo heart transplantation after several years. When they examined the explants for rejection, they found that antibody-mediated rejection was notable in almost half the cases, and it was previously unrecognized in 40%. And really, subclinical antibody-mediated rejection was noted to have occurred up to 4.5 years prior to the onset of graft failure. And when they looked at donor-specific antibodies in these groups, almost universally they had DSA at the time of graft failure, and most of these patients had class 2 antibodies at a median MFI of 5,000. So clearly, donor-specific antibodies that persist chronically cause even low-level allograft rejection does lead to graft failure. What about complement fixing antibodies? We heard a little bit about the ability to look at antibodies not just by MFI strength, but also the ability to fix complement in vitro, or using an assay to look for the ability for an antibody to bind complement. And we see that in this study, looking at all solid organ transplants, a meta-analysis of 37 studies involving almost 8,000 transplants, almost universally, the presence of complement binding antibodies was associated with increased risk of graft loss. And this included data from 370 heart transplants and 116 lung transplants. Well, clearly antibodies are deleterious to graft outcomes. What do we do when we see antibodies? Over the years, there have been a number of approaches to trying to reduce antibodies to improve outcomes following transplantation. These strategies include antibody removal with therapeutic plasma exchange, immunoabsorption, strategies to modulate B cells and plasma cell populations using CD20 antibodies or proteasome inhibitors, immunomodulation using intravenous immune globulin and tocilizumab, an interleukin-6 receptor antagonist, which I'll speak about a little bit. And also recently, we've published some data on using complement blockade with eculizumab. This is our earlier data showing that desensitization with plasma exchange, intravenous immune globulin, and rituximab certainly is effective, although the response is highly variable, with some patients not responding at all, some patients having a good response. But with this strategy, we are able to get most patients to transplant. And when we do get them to transplant, 5-year survival is really comparable to patients who are not sensitized, although there is an increased risk of antibody-mediated rejection and any treated rejection within the first few months after transplant. And most of these cases are easily treatable. When we looked at the use of proteasome inhibitors to knock out plasma cells, we see that, again, that 1-year outcomes are actually very favorable in this cohort, although we do see this early signal for antibody-mediated rejection within the first month or two after transplantation. And in all these cases, these episodes are eminently treatable. So desensitization certainly is a tool that can be used in highly sensitized patients to facilitate transplantation. What about some newer strategies? Well, tocilizumab is an interesting drug which blocks interleukin-6, which really is a master regulator of both T cell and B cell activity. It is involved in immune tolerance and with the regulatory T cells. It has a role in autoimmunity, allergy, the development of cytotoxic T cells, and also differentiation of plasma blasts into B cells and plasma cells. So it makes sense that by modulating this very important regulatory cytokines, we may have a significant impact on altering outcomes following transplantation. And certainly our colleagues in renal transplantation at Cedars-Sinai have been using tocilizumab for desensitization purposes, and they showed that they were able to actually significantly reduce immunodominant DSA in patients awaiting renal transplantation. And when these patients underwent heart transplantation, DSAs were really eliminated in all but one patient who really ended up with 2-week DSAs at 12 months after transplant, but no antibody-mediated rejection on protocol biopsy. So this is a very promising strategy for desensitization, which we're increasingly using at Cedars-Sinai. Well, what about complement inhibition? We know that we can use terminal complement inhibition with eculizumab, which essentially binds the complement cascade, blocks the complement cascade at a lower level so that it prevents the development of C5a and C5b anaphylotoxins and the membrane attack complex, which is deemed to be the most deleterious component of complement activation. Well, we've done a pilot study looking at the use of eculizumab at the time of transplant, with the first dose given in the operating room and subsequently for 2 months afterwards. And given that this is a highly sensitized cohort, we used a control matched population from the Paris transplant group. In Paris, the main strategy for transplanting highly sensitized patients is to use a combination of plasmapheresis and IVIG. And you can see the characteristics of our population is very, very highly sensitized. CPRA in our cohort was 95%, 75% in the Paris cohort. All the patients had a DSA greater than 5,000 MFI. Three of the patients actually had a positive CDC cross-match as well. And all of these patients had a positive flow cross-match for both T and B cells. What were the outcomes? Well, eculizumab was quite effective at blocking the development of antibody-mediated rejection, with only 25% developing this. The patients undergoing plasmapheresis and IVIG therapy at the time of transplant had much higher rates of antibody-mediated rejection. Importantly, post-transplant survival was excellent in both cohorts at around 90%. We also interestingly saw that antibodies actually started coming down after transplant in both groups, and that if you were able to deal with the antibodies early post-transplant, even though therapy was only for 2 months, these patients actually did well in terms of reducing antibody levels. And although the cohorts are very small in terms of looking at allograft vasculopathy, we really saw no signal for increased allograft vasculopathy, at least at 1 year. And so our desensitization protocols at Cedars really have evolved, and we will consider perioperative strategies in patients who are very sick, awaiting transplant on temporary mechanical devices. We'll use plasma cell therapies and plasmapheresis in more stable patients awaiting in-house, and then more chronic therapies with rituximab and tocilizumab in patients waiting from home for transplantation. One final thing I just wanted to let you share with you is some newer strategies that we're looking at to see if we can get some highly sensitized patients to transplant. Not all high-level antibodies or even complement binding antibodies are necessarily cytotoxic, and we are finding this out increasingly. And for some patients who have very high levels of complement binding antibodies with a very high CPRA, what we're increasingly doing now is to do what's called a surrogate cross-match. So we'll take their serum and we will perform a cell-based cytotoxic cross-match using a number of donors with corresponding antigens to determine whether those antibodies are truly cytotoxic. And you can see an example here where a recipient has antibodies to DQ4, 5, and 6, and using a variety of donors, we can deduce that the cell lysis occurs with all the antigens except for DQ4. And so in this particular patient, DQ4 is not a cytotoxic antibody, and we can cross that antibody at the time of transplant. And by using this strategy, we've been able to reduce the CPRAs for our patients awaiting transplant. We've been able to transplant 6 out of 9 patients using this strategy without having any rejection or graft dysfunction. 4 of these patients actually had C1q binding antibody, which we were able to remove from avoids. And we were able to cross C1q binding DSA in 3 out of 4 patients at the time of transplant. And in 2 of 9 patients, patients had a positive surrogate cross-match, and therefore there were no change in the voids. So this is another interesting strategy to use at the time of transplant for very highly sensitized patients. So in summary, allosensitization remains a major barrier for heart transplantation. Persistent de novo DSA and C1q binding antibodies are deleterious, but not all antibodies demonstrate potential for graft injury. Desensitization therapies demonstrate a variable response, but allow patients to proceed to transplantation with acceptable long-term outcomes despite increased risk of AMR. And perioperative strategies, including plasmapheresis, intravenous immune globulin, eculizumab, offer promise for crossing DSA with acceptable outcomes. And I'll stop there. Thank you for your attention.
Thank you very much, Dr. Jignesh. Very articulate and excellent presentation. Thank you. Now, just go on to invite Dr. Guillaume Coutons, who is transplant cardiologist in the very well-known La Salpêtrière Hospital in Paris, and he's part of the Paris Transplant Group, has multiple research publications in cardiac allograft, vasculopathy, AMR, and all the studies that Dr. Jignesh had mentioned. Welcome, Dr. Guillaume. Welcome from France.
Thank you very much for the introduction, and thank you very much for the invitation and the possibility that you offer me to discuss with you about our trajectory-based approach to cardiac allograft vasculopathy. It's a great pleasure for me to be virtually in front of you tonight. So let's talk about cardiac allograft vasculopathy. As you know all, it's a major cause of graft loss and patient death, but previous epidemiological approach were limited by different factors, and particularly by the binary gradation of CAV. We all know that CAV is absolutely not a yes or no disease, but is clearly a continuum spectrum of disease, and it was important to take into account this evolution. Previous study was also limited by their cross-sectional approach or their limited database. So our
Dr. Coutons, sorry to interrupt, you'll have to make it full screen.
Sorry?
You'll have to make your slides full screen.
Okay.
Thank you.
Sorry for this. So our main hypothesis was that we could be able to identify different profiles, different trajectories of evolution of cardiac allograft vasculopathy following our transplantation. We therefore performed a multi-center international study, including patients from La Salpêtrière Hospital and Georges Pompidou Hospital in Paris, patients from Leuven, and also patients from Cedars-Sinai in Los Angeles. We included all patients above 80 years old who had at least 2 cardiac allograft vasculopathy measurements after heart transplantation, for a total of 1,300 patients. The design of the study was quite simple. We collected all the data concerning the cardiac allograft vasculopathy beyond the first year post-transplant, and we analyzed different risk factors, both at transplant, during the first year post-transplant, and at the evaluation 1 year after our transplantation. You've got here the different data concerning the coronary angiogram for our study. with more than 4,000 coronary angiogram and a mean follow-up above 5 years post-transplant. As expected, the vast majority of coronary angiogram were strictly normal. We applied a latent class mixed model. It's a particular model, an unsupervised method adapted to the analysis of repeated measurements, and it is particularly useful to identify underlying groups in longitudinal data. As you can see here, our colleagues from the Paris transplant group just applied this LCMM approach to the EGFR following kidney transplantation. You've got here all individual trajectories of thousands of patients, and the LCMM will be able to identify distinct groups with similar trajectories or evolutive profile of EGFR after kidney transplantation. And we applied exactly the same methodology to CAV after heart transplantation. So let's talk about baseline characteristics of patients in our study. As expected, there were differences between our cohorts, and particularly you can see older recipients at Cedars-Sinai, and as expected, older donors in Europe, particularly in France, where we've got the median age of the donor was 45 years old, and for now, at La Pitié, we're about 50 years old of median donor age. So very old donors in Europe. And this will be very important to analyze the differences between the two cohorts of the European cohort and the American cohort concerning our results. For the identification of independent risk factors for cardiac vasculopathy, we analyzed a lot of parameters concerning the donors, the recipients, and particularly the dyslipidemia at 1 year post-transplant. The immunology with the presence of anti-HLA DSA at transplant or during the first year post-transplant, concerning about 1/4 of our patients, and also a lot of histological data because we've got almost 13,000 endometrial biopsy for the European study. So the main finding of our study was the identification of 4 distinct trajectories of cardiac vasculopathy following heart transplantation, going from trajectory 1 with no cardiac vasculopathy at baseline or during follow-up. The trajectory 2 or latent class 2, where you can see a mild and late onset of CAV after transplantation. Trajectory 3 with an early onset of cardiac vasculopathy, and trajectory 4, the most severe form of evolutive profile of CAV, with early and rapid onset and rapid deterioration of CAV after transplantation. Interestingly, we've been able to validate these 4 trajectories, this 4 evolutive profile in the independent American cohort from Cedars-Sinai with the same evolutive profile. The big difference was the proportion of patients in each class in each latent class. In Europe, there was only 56% of patients with latent class 1 without any CAV during follow-up, compared to 3/4 of patients at Cedars. And it was completely different for latent class 3 with twice more latent class 4 in Europe compared to the US. And the most important risk factor is donor age, and was particularly different between our 2 cohorts. Interestingly, when we pool all this data with the 1,300 patients, you can see that these trajectories are associated with distant prognosis after transplantation. It's not obvious for trajectory 1 and 2, but clearly trajectory 4, 3, and particularly 4 are associated with very poor post-transplant outcomes and early mortality. Now, what about risk factors associated with the progression and the cardiac vasculopathy? You can see all what we analyzed and what was significant in univariable analysis. I will not going to go into details of all this, And I will go directly to the multivariable model, where we can see that we identified 6 independent predictive variables associated with the different trajectories. Taking trajectory 1 as the reference, you can see that donor age, donor gender, donor smoking, dyslipidemia at 1 year post-transplant, and 2 important immunological parameters were significantly associated with our trajectory. Particularly, you've got the class 2 anti-HLA antibody during the first year post-transplant, either preform or de novo during the first year post-transplant, were independently associated with the latent class 3. And the acute cellular rejection greater or equal than 2R during the first year was associated with our latent class 2. So as a conclusion, we identified 4 trajectories of CAV after our transplantation. These trajectories were validated in an independent American cohort. The 4 trajectories were associated with distinct prognosis and determinants, and particularly donor-related risk factors, metabolic profile with dyslipidemia at 1 year, and an immunologic profile with anti-HLA DSA, class 2 anti-HLA DSA, and acute cellular rejection. And our study provides a basis for trajectory-based assessment after transplantation that may enhance risk stratification, individual risk stratification, and we developed an online tool, which is in open access, to evaluate individually the risk of progression of CAV for each patient that you may see in your outpatient visit. And finally, you can find all the details of this presentation in our article published last year in Circulation. I would like to thank again the organizer of this very interesting meeting, and to thank all our colleagues who participated actively in this research, and particularly colleagues from Cedars-Sinai. Thank you very much for your attention.
Thank you, Dr. Guillaume. Very pleased, very nice presentation, and very useful.
Thank you.
Now, if we go to, we can ask and take some questions. So one of them is from Dr. Miran. So maybe Dr. Jignesh Patel, you can answer. So he says that if it is a high CPRA and positive virtual cross match, and there is no negative flow cross match, and there is a negative flow cross match, so would you desensitize?
If you have the question is relating to a flow cross match at the time of transplant, or
yeah, that probably is asking at the time of transplant, just before transplant, high CPRA cross match, but the flow cross match is negative. So would you...
If the flow cross match is negative, then we would just generally transplant this patient with routine induction therapy with anti-thymocyte globulin, and go ahead with that. It really, we actually transplant across positive flow cross matches not infrequently, because sensitization, usually patients who are very highly sensitized will have a positive flow cross match, but a negative CDC cross match as the most common presentation. But if a negative flow cross match is actually a very safe patient to transplant with routine induction therapy.
Right. So other one would be if Dr. Srinivasan is there. Srinivasan, are you there?
Yes, I am.
Yes. So yes, since you are in the discussion, you have seen, what do you think is the possibility of a nationwide CPRA calculator? I mean, deriving such a system. Is it, you think it is feasible or possible?
I thank you very much for inviting me to be here. I think whenever there are challenges, I really get excited. My adrenaline goes up. There are 4 things which are brought out today, and all 4 have solutions. The first one is the repeatedly emphasize the need for a high resolution HLA typing. As you know pretty well, we have an EFI accredited laboratory offering the NGS HLA. And the second is specifically the CPRA, which we did discuss earlier. The challenge is, one, the data for the HLA frequencies of Indian population. And we do know from published 6 published articles in peer-reviewed journals from our own registry that the distribution, the top 20 HLA alleles are different in 6 linguistic populations, even within India. So we have an ethnically variant situation, but we have a data of close to 15,000 plus stem cell donors. One of the speakers did emphasize the data from stem cell donor registry is far better than what came from the renal transplant database. So we do have the data, and I'm happy to say that the discussions have already started in the background, and I'm pretty confident in the next couple of months we will have answer to the CPRA with the Indian data. So that is something which I'm pretty confident now. And the third point which was brought out is the extension of the divergent HLA frequencies that we have in Indian population. What Dolly was telling about the need for the 50% of the beads, 50% of the antigens are not represented in the beads of the 2 vendors. I think here is the request to Dr. Professor K.R. Balakrishnan that I know that your interest in research. I think it is time we put our heads together seeking the support from any one of the institutions to develop our own beads with the material that is available within India. It's not just going to help India, it's going to help Indians globally. I think it is more important. And from whatever data we have seen that the HLA distribution here is sort of more or less similar to the South Asian population. So we are talking about a sort of a domino effect. What we are going to do here is going to touch the SAARC region or people from the SAARC region. And the last is there is a compelling need to have a patient database with high resolution HLA typing and possibly baseline DSA testing data, and look for the ideal patient when there is a potential donor. This is something is again doable pretty quickly because over the last 6 years, we have developed our own version of the search database where the current database contains the stem cell donors. We put in a patient HLA to search, and we just need to flip it around, which won't take much time, plus include the DSA and the excluded, whatever the antibody if the donor patient is going to have that needs to be excluded. As I did listen to one of the earlier presentations, which was available on the net by Dr. Patel, that we don't want to be disturbed in the middle of the night when the DSA is something which is sitting there. We don't want to be called. So I think there are 4 challenges. All 4 could be addressed in a short time. All that we need to do is that we need to put our heads together, and probably we could take it offline. On the question on the specific, Dr. Ravi Kumar, you said national. Over the last, sorry if I'm going overboard here, over the last 12 years with 3 governments and several secretaries and health ministers, I've been hitting Delhi for the National Bone Marrow Donor Registry, and we have not succeeded. I know somebody from NATO is sitting here, and my suggestion will be to be local, regional, develop a model, pilot it, and then tell your neighbors and colleagues that, look, this can be laterally expanded and it is feasible to do. All these things quickly before we see the 31st of December of 2021, it's possible. I'm a born optimist. I'm available at any time to drop in and have the discussions. Thanks.
And Professor Dital, Kumud Dital, or Dr. Jignesh, Dr. Sandeep said, any comments? Go ahead.
Oh, listen, comments more in terms of the practicality of what we're doing here. As you know, I've just arrived 3 months ago, and I'm surprised that biopsies and surveillance biopsies were not done after heart transplantation. And I'm trying to be religious about it, and the DSAs afterwards. But I'm finding it difficult because it is expensive even then, and it's not routinely done. So we go back to Dr. Kim's talk, where there may be hope in terms of using a non-invasive tool, for example, in terms of limiting the endomyocardial biopsies, certainly in the first year. But I was surprised again that with all the knowledge that we have, and a lot of the trials having already been done before 2015, is that the example Dr. Kim quoted, and this is also a question for Dr. Kim if he's around, was one of a patient in whom it was difficult to do biopsies by virtue of the venous state of the veins. And so my question is, and they're still doing endomyocardial biopsies. So if they're in these sort of centers where resources aren't such a big constraint as here, and I haven't even gone into broach the subject of cost, that if they're not yet doing this, they don't feel confident enough to take this on board, then, you know, it's for us, it's a really huge milestone to try and achieve. And so what are our options here? I'm really interested initially in trying to get them through a safe passage in the first year, primarily on trying to match them appropriately and the surveillance. The issue of antibody-mediated rejection, which we know is about 10% later on, is a crucial figure, but then how we deal with that is a secondary issue. And I don't think we're still there. I think at the moment we're still calculating PRA. The regulatory authorities require you to register PRA. I don't even know whether if you calculated a PRA, they will be acceptable, because that is done by purely a mathematical tool, as we've heard. So, and the authorities are very strict about having something that they can't read between the lines, and therefore it must be absolutely apparent, the technology and how it's reported. And so we have a big hurdle. And I like Dr. Srinivasa's optimism. I'd love to share it and have further discussions with him. But maybe Dr. Kim could tell us, and maybe even Jignesh could tell us, what's happened, when do they think that they can replace their endomyocardial biopsies with AlloMap or non-invasive tools? I know there are other things coming on into microRNAs and so on, but just with what we have with AlloMap, you know, it's one thing to do it for the exceptional patient. I'm talking about the routine patients, and when will that happen?
I don't know if Dr. Kim's still on, but I can tell you in our program, we have really pivoted away from invasive methods for surveillance. And in our program, we're actually only biopsying in the first 2 months because the AlloMap test is really not validated in the first 55 days. Yeah. So, and that's in the first 2 months is when you see most of the rejection. And so following that period, we're transitioning to the AlloMap, AlloSure technologies with both the AlloMap and the cell-free DNA for post-transplant surveillance. The high-risk groups, we're still biopsying because we do get the very highly sensitized patients that we're transplanting with eculizumab, for example. Those patients will still biopsy because the AMR rates are quite high in that cohort, and we do want to know that data. Also, we also know that, and I'll show you this data tomorrow, that patients who have antibodies actually have high basal levels of cell-free DNA. So that test becomes less helpful when you see an individual number in an individual patient. I also want to emphasize the importance of Dr. Coutance's presentation, which has really pivoted away from doing surveillance angiographies. And the importance of that study is that using this unsupervised machine learning algorithm, we were able to come up with a calculator that was validated in very different transplant programs across 2 different continents, where transplantation takes place with different protocols, different donor ages, different post-transplant immunosuppressive regimens, as you saw with our eculizumab and plasmapheresis protocols between the 2. Nonetheless, this calculator has enabled us to reduce the number of surveillance angiograms during this COVID period by more than 75%. And so even with angiographies, we're now moving away from doing routine procedures. So I think the outlook is very bright for monitoring patients without the need for expensive invasive procedures post-transplant.
I mean, I agree with that, Jignesh, but my worry is, as your program, and certainly the ones that I was working in Australia and in the UK, half your patient list is with an in situ VAD. And so while I agree that this may be possible, you're also saying that for the highly sensitized and the complex patients, you're still doing the biopsy. So the question really comes down, what fraction of your patients can you actually do this with? And how many of your patients are still doing biopsies on? Because the complex patients are forever increasing.
Yeah, so right now, about 80% of our patients are routine patients, and it's only 20% of the patients that require the closer evaluation. Okay. So the vast majority of patients we're able to transition away from biopsies.
And finally, the cost?
We actually don't biopsy after the first year, and we haven't biopsied after the first year for the last 10 years.
I think that's true for most centers. But what about the cost?
So the cost of the AlloMap test in the United States is still not insignificant. It's still a fairly expensive test. Almost, but it's still cheaper than doing an endomyocardial biopsy when you look at the total cost of doing a biopsy. We don't know what the cost for cell-free DNA will be. It's available through a protocol right now. But I suspect that the way it's going to be marketed by CareDx will be a dual test available called HeartCare, where they provide both an AlloMap and a cell-free DNA score together. But we'll wait and see what those costs are.
Sorry to labor the point, but for my colleagues here, it is important to know what that relative expense is in dollar value, because your endomyocardial biopsies may cost significantly more than in India, and therefore, on balance, your test may be very much more, unreachable for us. So,
yeah. So, in the United States, cost is always an issue. The endomyocardial biopsy, for example, is about $35,000.
Yeah.
In contrast, the AlloMap test is about $3,500.
Yeah.
So there's still a significant cost benefit in terms of going to the non-invasive strategy, but the $3,500 is still arguably a much higher cost for a non-invasive test than it should be. Now, the cell-free DNA may actually be a lot cheaper simply because there are multiple platforms that are emerging. And so there may be competition in that market that will allow us to actually get a much cheaper test. And cell-free DNA is a technology that really can be developed even in-house. So it doesn't really require a proprietary technology.
My last comment, Ravi, I apologize, but your cost of endomyocardial biopsy is almost exceeding the cost of a heart transplant in India.
Yeah, I realize that.
If I may add, one concern from our experience is that both the AlloMap and the donor-derived cell-free DNA, presently for our institution, we have probably a 60% rate of multi-organ transplants. And the utility of these testing for multi-organ transplants is yet to be defined. So that's another factor that makes us rely more on endomyocardial biopsies for surveillance.
Thank you. Dr. Kumut, one point that you raised about EMB. So here in Dr. Bala's unit, last 470 heart transplants. Yes. We have done EM biopsy for every case. We have the data, we just got to publish, but we are doing it 4 times per year. First year. Yes. But after first year, I mean, we have been doing it every year with angiogram.
Yes.
So as Dr. Patel said, I don't know, maybe as they're given a moved away from the post 1 year EMB, but we have been doing it. I don't know, as a probably old, whatever.
I think that's a sensible attitude, which is to do it. The first year is crucial. In fact, most would say the first 6 months is the crucial, and that you're doing maybe weekly for the first month and once monthly up to 4 to 6 months. After that, I'm not sure whether you really have to do it, but that's a really crucial period. So when you take that into the reality of what is achievable with cost and what patients here can afford, because that's the absolute bottom line, then it seems that for the cost of the AlloMap is your cost of the biopsy. Your cost of the biopsy in the States is the cost of the transplant. So if you added a secondary non-invasive test, it would go way beyond. And so I think we just have to be careful of what we can do in this context we have to search our own ways of non-invasively finding a way to replace these endomyocardial biopsies. And I know there's a risk, but the risk is very fine. But I think on balance, if you leave it for at least 6 months or a year, those risks can be mitigated by the fact that you're saving lives and certainly safeguarding graft and the recipient survival to medium and long term.
So any comments from Dr. Sarvesh Paul or Sandeep Seth? Dr. Keval, would you like to add?
I think Dr. Seth has maybe left.
Yeah. Can you?
So we religiously do biopsies.
In names,
like Dr. Dittal is mentioning, we religiously do biopsies, first within 15 days, then 1 month, then 3 months, 6 months. So I think we are the only institute who is doing regular biopsies in heart transplants. And yes, whenever there is a suspicion for rejection. My one question to Dr. Jignesh Patel would be, if he's there, still there. Dr. Jignesh is there?
Yes. Yeah.
Hello, sir. Good morning to you. When we are talking about these non-invasive tests, like Dr. Dittal has mentioned that it involves a significant expenditure. Yours is an institute which does a number of transplants. Dr. Bala does a number of transplants. India is a diverse country in which not many institutes are doing maybe more than 20 transplants per year. So actually, if you want to go for this, definitely installing it in an institute like ours is a government institute is fine. We can do that. But it's not possible for everyone. So in your opinion, what should be an average volume for an institute to probably start a non-invasive testing, like AlloMap or maybe cell-free DNA?
Yeah, I think it's a good question. It's a question of what the resources are for establishing that technology. I think with cell-free DNA, that would be fairly straightforward to do because you just need a lab that is able to sequence and establish the platform. And remember, cell-free DNA is being validated for all solid organ transplants. And so you may have a kidney transplant program or a liver program or lung program, and you could use the same technology for all those organs as well. And I'll be showing some data tomorrow regarding cell-free DNA. In fact, Dr. Shah's group from the Graft Consortium just published in Circulation some very, very interesting data from one of the largest cohorts using cell-free DNA, showing that this technology is really going to be a game changer in terms of our ability to not only diagnose rejection, but also predict rejection. We also forget that although the endomyocardial biopsy is a gold standard, it is a very imperfect gold standard. There is a lot of issues regarding reading of the pathology. The concordance rate amongst pathologists is quite low. And so we really do need to move away from the endomyocardial biopsy and have some more sophisticated methods for looking for allograft rejection. And I think one of the things about non-invasive testing is not just because it's going to be cheaper and more and better for the patient, but the hope is that it's actually going to be a superior technology so that we can prevent allograft rejection in these patients. And I think the cell-free DNA platform is going to be very promising for places where transplants are emerging, even in small centers. AlloMap is very involved. It requires a central lab. It requires the sample to get to that central lab in a certain period of time. And there's a proprietary technology that CareDx kind of sits on and doesn't share. And so unless CareDx is going to set up a lab in India or they're going to facilitate that across the continent, I cannot see that being something that's going to be routinely available or easily available there for some time.
Dr. Jignesh, I'd like to invite Professor Jasmeet Kaur. She represents a very big lab called Lalpath. So what are your comments of Jasmeet about cell-free DNA? Your lab is probably developing.
So I just wanted to add to what Dr. Patel said. We at Dr. Lalpath Lab have partnered with KDX, and I'm in the process of validating cell-free DNA, and I think I'll be launching the test in a week or two. I will briefly, of course, I don't have the experience beyond validation, but I will be talking about it tomorrow. So it's not that we can't do it. It's a very easy test to perform. It's on the NGS platform. Yes, it does need a little initial investment, but I think we, with our entire range of antibody tests that we are doing with the single antigen B and the C1q, should be able to support all the transplant organs. We're already validating for kidney. I've in touch with Dr. Ravi Kumar for the heart samples, and I think we should, AlloMap, yes, like Dr. Patel said, not yet. It does require some different, partnership with the KDX, but the cell-free DNA is as good as on from Feb in India. So I just wanted to add that little comment there.
Thank you, Jasmine. Now, just one question to Dr. Jignesh that's come in the Q&A. What do you think are the importance of non-HLA antibodies for? I think the question was for lungs, but for heart and what is it from your side?
Yeah, it is an increasingly difficult area to look at. This is an evolving area. We're finding more and more non-HLA antigens that may be to which the development of antibodies has a significant impact on graft function. We have an expanding panel of non-HLA antibodies that we look at in patients that do present with rejection. We've routinely looked at, for example, AT1R antibodies in our population, and we have quite a lot of data on that. But there's a lot of data on anti-endothelial cell antibodies, MIC-A and MIC-B antibodies as well, that may have a role in causing graft dysfunction. The problem with this area is that the tests are not validated generally. Antibodies can be frequently found when there is no allograft rejection, so we don't really know what the precise role of these antibodies is when we see it. But we certainly have a cohort of patients that have had rejection due to non-HLA antigens, to which they've responded with appropriate therapies with a decline in those non-HLAs. So it is something that occurs on occasion, and you do have to be cognizant of the fact that the patient may have this going on.
Any other comment, Dr. Keval, anything you'd like to add? You're muted, sir. You're muted.
Yeah, the biopsies are meant for this to check the rejection, but also to reduce the dose of immunosuppression. Have you seen a significant reduction in the doses by the number of biopsies, or it has remained same in last 5-6 years, and renal toxicity is same? Because biopsies are also related, they also have the complications of causing TR and stuff. So
yeah, we don't really use biopsies to adjust immunosuppression. Our immunosuppression protocols really are based on therapeutic drug monitoring. We do use, on occasion, when patients have complications such as infection and malignancy, we'll also use technologies such as the T-cell immune function assay to look at T-cell activity. But in general, we will use therapeutic drug monitoring to guide our immunosuppression, and really the biopsies are to look for early rejection prior to any evidence of graft dysfunction when it's under a surveillance protocol. So that's really where the utility of the biopsy is. The other thing is that in these days, the amount of rejection that we're seeing is actually quite low, which makes doing studies quite difficult. For example, in Dr. Shah's study, I think the rejection rates were about 5% in that cohort. And so we're not seeing it that often because the immunosuppressive regimens are now so good, and our ability to monitor levels and keep immunosuppression where it needs to be is very good. We're only actually seeing rejection when there is some perturbation of the immunosuppression in patients, and that's when they get into trouble, particularly when they develop this late onset antibody and late AMR. It usually occurs because there's been some interruption of immunosuppression.
Ravi, you may make a comment on that. Interesting comment, Keval. I mean, I think Jignesh pointed out earlier the issues and problems with endomyocardial biopsies, and I'll readily admit the fact that you can be taking out scarred tissue from previous biopsies. There is a inter-observer variation amongst pathologists, all of that. But I think it would be dangerous to use that to monitor, to tailor immunosuppression. At the same time, what you raise is a really important problem in India, where because of the inherently greater risk, at least perceived risk of infection, there is a great wish to try and reduce the immunosuppression level. And from everything that you said, Jignesh, and certainly from the results, I'm sure the others would say the same, is that the low level of rejection that we're picking up probably suggests that for the heart at least, across the globe, we're probably over immunosuppressing. And I think if we can get together in India and collaborate, we'll probably come up with a much better protocol in terms of how to effectively do that. That would be of benefit across the oceans. That's certainly a project that I think would be worth, Ravi, for us to try and tackle with.
In this, Dr. Jignesh, in that connection, one question to you. The Paris transplant group, I think, developed a system called iBox for kidney. I mean, collating all the. You think there's a scope for the heart?
Well, the vasculopathy project that we did is based on the iBox, looking at long-term allograft vasculopathy. It's the same technology that was adopted, and it's really the cardiac iBox that we used for that. Going back to infection risk and immunosuppression and doing analyses beyond therapeutic drug monitoring, I just wonder whether there would be a greater benefit for the use of technologies such as the T-cell immune function assay in India, because it's been pretty well validated that if you have a Silex score less than 100, that's associated with a fourfold hazard ratio for infection. And for us, it's been a useful technology in patients who have complex infections to be able to back off on their immunosuppression. And something of looking at T-cell function, antibody function may be useful in that sort of scenario. We look at immunoglobulin levels for hypogammaglobulinemia, for example, in infected patients, because there's some Brazilian data to show that IVIG supplementation in post-transplant infections is associated with a survival benefit. So those are areas looking at the other side of the coin in terms of T-cell activity and antibody activity may be helpful to minimize that infection risk.
I think Silex should be introduced, or in a larger scale, they can develop it. Any other comments from anybody?
I just wanted to add. Just wanted to add one point to what Dr. Srinivasan pointed earlier. In terms of the HLA profiles of the Indian population and to be able to develop the beads that we can cover all specificities, our data from our lab has indicated that Indian population carries alleles which some are novel and unique, but majority of them are Caucasians, but we do also represent the Oriental side of gene pool. So in fact, when we have the data from the Indian gene pool and we try to develop the beads, we will really be able to help globally, not only the Western part of the world, but the Eastern part of the world as well. And the same thing holds true when I talk to people in terms of the bone marrow registry, that if we have more volunteer donors, we could be serving the whole globe, and like Be the Match and, the German registries and the Anthony Nolans, all of them, when we compare our data, we have done extensive studies on HLA profiles in North Indian population and some tribals and some Central Indian populations. It does show that that's the advantage. And one remark or question I had to Dr. Patel. So you mentioned about this new assay that you're trying to, anticipate the immunogenicity using different lymphocyte. Is that the new assay that you're trying to develop? You said, are you going to be using a panel of fresh lymphocytes from individuals or something?
Well, the Silex assay is actually commercially available. It's been available for some years. And the way it works is that you take blood and you magnetically separate T cells and then treat the T cells with phytohemagglutinin and measure the ATP levels. And those ATP levels are a direct measure of T cell activity. And so this test actually has been validated in multiple solid organ transplants, showing it's particularly helpful when you have a low score below 100, because that's where the infection issue occurs. A high score has a loose correlation with rejection, but it's really not that well validated. We published on this a few years ago. But the low scores are particularly helpful, particularly in cases such as infection and malignancy. Patients with post-transplant malignancies, we're able to kind of back off on immunosuppression based on Silex as well.
Thank you.
Ravi, I've had a few questions come through, and the one that's uncovered is in the context of non-invasive techniques. And the question to Dr. Kim and Jignesh is, what about cardiac MRI? And when I was down in Sydney, Peter McDonald's group and Dr. Jabor there were certainly coming out with some very interesting MRI data. And there's certainly a trial going on at the moment, a prospective trial. What are you investigating that, and where does that sit in respect of AlloMap, AlloSure, cell-free DNAs, and so on?
Yeah, I don't know if Dr. Kim, is he still there, if he wants to comment on this?
Dr. Kim has left.
Okay. So yeah, MRI is we're finding it to be a very useful technique in determining allograft rejection, particularly in patients with recurrent rejection and persistent graft dysfunction. And what we're finding is that we're seeing signals for myocardial edema with increased T2 relaxation times. The use of gadolinium to look at myocardial scar helps us determine how much ongoing inflammation there is versus how much scar development there is. And it's particularly useful technology to determine whether it's safe enough for the patient to be relisted for transplant, where we think that the graft is not going to improve, particularly when there's a lot of scar tissue there. And when there is ongoing edema, that allows us to determine the need for ongoing therapies in some of these patients, particularly when they're, for example, persistent antibodies as well. We have a number of research protocols in this area ongoing with the newer MRI platforms as well.
Thank you.
So if there are no more questions, can we adjourn for the day? And I would request everyone to rejoin tomorrow, because there's some lot of interesting discussions on molecular microscopy. Dr. Patel is going to talk on cell-free DNA, Dr. Palaksha on miRNA and immune monitoring. So we will probably, there are some novel areas which we'll cover tomorrow. And two of the stalwarts who are joining us, Dr. Philippe Halloran and Dr. Alexandre Lupi from Paris. They are the acknowledged experts in this area of molecular microscopy. So I would request all to join tomorrow. So thank you.
We should develop some collaborative programs through this webinar, through
yes.
All the centers in India should join together, because of course the rejections are few, but if we put together the data and try and explore some new parameters, we will get some interesting findings.
So, Dr. Keval and Dr. Kumud can take the lead, and your Srinivas and PGA, Dr. Uman, so many, leading people are already here. So Keval,
That's why I put this request.
Happy to help.
Keval, anything like that?
Thank you so much. Thank you.
Bye-bye.
Good night. Great learning.
Dr. Keval, you are muted again.
So we'll make a circle and we'll ask one person to take a lead, and everybody would be connected to that key person, so that so the other people can connect, and one can guide the other people also. We can make a group, WhatsApp group, or some.
Yeah, we have from our institute, we have Dr. Sarvesh, Dr. Sandeep Seth. So it's going to be very good if they can lead, and then we can all join hands.
Yeah, for sure, we'll do that.
Thank you so much for that.
Thank you.
All right.
So if there are no more questions, we adjourn. Tomorrow we'll meet again at 7 p.m.
Okay.
Thank you. Thank you all. Thank you, Dr. Tigner. Thank you, Kim. Thank you.